Okay, welcome everybody. My name is Dr. Amanda Rosenzweig, and I am the Assistant Dean for the School of STEM at Delgado Community College. And at Delgado Community College, we do have a program called Science Laboratory Technology. And the SLT program is an Associate of Applied Science, and it offers students an exciting opportunity to gain hands-on experience and lab techniques while preparing for careers in biotechnology and chemical technology. So we often get a lot of questions from students, why should I choose SLT? So some of the answers are relatively simple. It's fun, but to go into a little bit more detail about it, I want to show you some of the information that we can provide you. We are an open admissions program. And what that means is students can easily enroll and begin their core courses. And those core courses are general biology, lecture and lab, as well as general chemistry, lecture and lab. Once those courses are complete, they will move into entry-level courses, which is our Introduction to Science Laboratory Technology. And that is offered every fall and spring. And once they take those courses, they will then be able to go through and complete the remaining curriculum, depending on which concentration they want, biotech or chemtech. And we often have students do a combination of both, or they actually complete their degree with both concentrations. Now, we have a lot of industry-related training. So our faculty often go to trainings held throughout the nation to learn specific techniques and current trends. And this is going to include biomanufacturing practices, monoclonal antibody research, and of course, biosafety techniques. We get a lot of real world experience also with our students. Our students will have a lot of emphasis on internships, laboratory research, and industry partnerships, and that helps ensures that they are job ready. And you can see a lot of our students do go on to a four-year university, but many of them go directly into the job field. We also have a lot of students do both, where they'll work full and part-time, as well as go on and pursue their bachelor's and master's. There is a very diverse career path within the greater New Orleans area or the Gulf South region, but we have students that do move on to different states. We have Minnesota, Arizona, New Mexico, and California that some of our students have moved to. But we have our students have secured positions at leading organizations, Chevron, Dow, USDA, Tulane School of Medicine. So we have a lot of community interest and support in our program, which helps. Earlier, I noted that we have two concentrations, biotechnology and chemical technology. Biotechnology is going to focus on molecular biology, biomanufacturing, biomedical research, cell culturing, and chemical technology is going to do analytical chemistry, instrumentation, and chemical processing. Again, there's overlap between both, and that's why a lot of our students do take both concentrations. We have tons of faculty expertise, and a lot of our faculty are on today. So thank you to our faculty who allow our students to be successful. They are bringing cutting edge research and experience in synthetic biology, molecular genetics, toxicology, bioanalytical chemistry. And as I said earlier, they continuously are trained in new and cutting edge techniques. We are very fortunate that we have grants that support us. So we have multiple different grants going on right now. We have a grant aligned with the USDA, the ARISE grant. We have a biomanufacturing practices grant that will be start to be incorporated in the fall semester. We do algae research that's supported by ATEC and SBI, which is a scientific instrumentation company. And then our Biosafety 2 laboratory is almost open. We're waiting for our final approval so we can do a little bit more intensive research. So just to go over a few of our relationships, we have relationships with many universities as well as many industry partners that have been phenomenally supportive. We are currently working on a potential pathway for the NOPD, the New Orleans Police Department, for students to go into criminal justice, forensics, and biotechnology, chem technology. So we're working on that right now. That's in the early stages, but lots of support from the community. And then we also have a lot of national engagement. So we work with a lot of consortiums and institutions around the nation that help support both faculty training and student opportunities. And then I'm not going to go over this, but the rest of this presentation is just going to introduce our faculty. And then we have a list of our some student testimonials, just a few of our student graduates. So this will be shared out with everyone, but I just wanted to kind of give you an introduction to what our program's about and the opportunities that have occurred within the program. Just a short background on the program is we started the program, we got the funding for the program in 2015. It fully went into effect in about 2016, 17. And then I became a part of the program in 2018. So we had a different team help bring the program to light. And then all the teachers and students and myself that have been involved since have continued to brand the program and grow it to what it is today. So does anyone have any questions about the program that either myself or one of the faculty members online would be able to answer? Okay. So the last thing I want to go over is just some, sorry, I didn't mean to do that to y'all. Just some housekeeping rules. The presentations are allotted 20 minutes each. That does include the questioning session. You can put questions in the chat. I'll be sure to be following the chat so I can ask the questions at the end, and that way the speaker does not need to keep track of the chat. If you have a question that you think is really relevant to the moment that the speaker is talking, put your hand up using the emoticons, and that way the speaker can call on you and answer your question as it's relevant to the topics being spoken. For our students, I want to make sure that your professors know that you have attended. So in the chat is going to be a short survey. It's 4 questions: name, email, who your professor is, and if you want more information. So please fill out that survey so you can get credit for attending. And then we have a lot of ways to get in contact with us for more information. We are very active on social media, LinkedIn, Instagram, Facebook. And then depending on your goals, there are different individuals that you can contact. And then last, I'm just going to scroll up one more slide. This QR code should take you to the SLT homepage, where you can dig deep into information on the program. Now, one of our directors is online today, and I just want to give Ms. Charlene Shunick a short period just to welcome everybody. Dr. April Noble is unable to attend today, but she is our Chem Tech director, and she's had tons of success with our students, who some of them I've seen online today also. But Charlie, do you want to take it away for a second?
My name is Charlie Shunick. I'm the biotechnology director within the SLT program, and I've been in this position around when Amanda came in since 2018. It's been really fun to watch the program grow. My primary role is to make sure our students are happy, make sure our teachers are happy, encourage internship opportunities, and ensure that students are getting into the places that they need in order to reach their goals. And also just make sure that we're tracking what our students are doing after the program. So it's been really fun. We have a great team of teachers and a lot of opportunities that keep happening in New Orleans. It's been amazing.
Okay, thank you, Charlie. So I am really excited because we have curated a phenomenal group of speakers today, some of which I have close personal relationships with, and I have the honor of introducing the first speaker, Dr. Jim DeKloe, or James DeKloe. I've known Jim for, I guess, my full 8 years within the SLT program. He's been a great mentor and friend to me. But Jim is a distinguished professor of biological and biotechnology at Solano Community College in California. And he founded and directs the Industrial Biotechnology Program, which has significantly contributed to the workforce development in that sector of California. But I do want to say that something that you won't find online about Jim is that we all call him the godfather of biotechnology, and it's his effort to support students' success in this field is uncanny, and we're just lucky to have him today speaking, as well as a supporter of the Delgado program also. So Jim, I'm going to stop sharing, and if you have a presentation, it should allow you to directly share yourself.
I do. Thanks,
Okay.
Amanda. Right. Can you see? Okay. Thank you. And I started my career as a chem tech, and that is a worthy career. But today I'm going to talk about biotechnology, and I have a presentation called Microbes, Medicine, and Money. So I'm going to try to convince you that biotechnology is the most important topic in the world, and that it is going to fundamentally change the human condition even more than it already has. So this is the biotechnology century, and Freeman Dyson was a physicist, well-respected, and a futurist. He said the 20th century was the century of physics. The 21st century will be the century of biology. Biology is now bigger than physics as measured by the size of the budgets, the size of the workforce, and the output of major discoveries. It is likely that biotechnology will dominate our lives and our economic activities in the second half of the 21st century, the way that computer technology dominated our lives and our economy in the second half of the 20th century. And every year I go to this conference called SynBio Beta. I love this saying, biologize the future. And every time I'm at that conference, I think, wow, I'm at Macworld 1984. That is, I'm looking into computers right before the internet hits and right after Macintosh has been introduced. So what is biotechnology? Biotechnology uses living cells to make products. It takes something, a raw material, ground up grapes, and then you add a living cell. In the case of making wine, it's a yeast. And then my neighboring county of Napa takes grape juice, adds yeast, and that provides a significant boost in its value. So you take about $1 worth of grape juice. Actually, it's a couple of dollars because, oh, they have a fancy French name. Grapes are Cabernet Sauvignon or Sauvignon Blanc. And you add yeast and slap a fancy label on it. And that grape juice worth a few dollars now is worth $30, or a fancy enough label, a couple of $100. So this is the old definition of biotechnology, meaning that this is nothing new. It's been with us for a long time. Now what we really mean is the use of genetically engineered organisms to produce pharmaceutically active proteins.
Hey, Jim,
So we
I'm sorry to interrupt. Your slides aren't advancing.
my slides aren't advancing. Okay. They were advancing for me. They looked great. Thanks. Let me try again. Thanks, Amanda. Can you see it now, Amanda?
It's
You, bud?
still on the preview mode. It's not showing full slide mode yet.
Okay. Can you see the slide though?
Yeah, we can see your slides. Like right now, it's on the slide 8, new biotechnology.
Yes.
Okay.
Good. Okay. Right. I'm thinking you can preview what I'm going to talk about next, and I think I'm, in the case of time, just going to continue. So this is a product made across the street from me. This is Herceptin, and Herceptin is an antibody that attacks a especially aggressive type of breast cancer. The women before this, the development of this product, were told by their physician, you have less than a year to live. With the injection of this antibody, and the antibody goes to the bloodstream and attaches to a protein on the surface of the cancer cells. With this, the women are living beyond the 5-year mark, which is the endpoint. And so these are game changers. And I want to convince you that not only is this one of the biggest industry in the world now, but it will be in the future. So it's not only science, it's engineering, medicine. Important in this is government regulation, especially by the Food and Drug Administration. Business is important because you need to make a profit and keep the doors of your company open. And so that is all of these fields intersect. And I always tell business majors that if you are agnostic about what business you would like to go into, I think that you'd be foolish to go into anything but pharmaceuticals, because that's where the real money is. Okay, so this came out of the 70s, and laboratory development of genetically engineered cells, the ability to put human DNA into cells of other types, either bacteria or yeast or mammalian cells, now algal cells and other types of cells. And that was a game changer. And it all starts with not a joke. Two guys walk into a bar. So in the mid 1970s, the guy on the left, named Bob Swanson, was a 28-year-old recent graduate of MIT's business school, and he started calling up scientists and said, "Do you want to start a new company?" And yeah, scientists all said, "I'm a science guy. I'm really not into a company." And he said, and then he called Herb Boyer at the University of California, San Francisco, and he said, "I have access to money." And Herb Boyer said, "Oh, my laboratory always needs money. So yeah, let's have a beer. I'll give you 15 minutes. And the 15 minutes turned into 3 hours. And at the end of the 3 hours, they were writing on the back of an envelope, genetic engineering technology, Genentech. And that was the first biotech company. And it founded itself over a glass of beer, which of course is ancient biotechnology. So there is a bronze statue on the main campus right outside of the research building of Genentech. And that immortalizes that meeting in a bar that started the multi-trillion-dollar industry of biotechnology. Of course, you needed funding, and the wisdom at the time was no one will ever found another pharmaceutical company. The barrier to entry is just too great. The barrier to entry, it just takes too much cash. And they went to Kleiner and Perkins, and the venture capitalists that founded Silicon Valley, and they said, "We've got something that's risky." And they said, "Well, Do you think you can do it? Do you think you can make a pharmaceutical company based on making the medicines using living cells rather than chemical reactions in a tank? Yes, we think we can do it. Are you sure you can do it? No, we're not sure we can do it. But do you think you can do it? Yeah. Well, what the heck? I gave money to these two smelly guys who were making computers in their garage, and that turned out to be Apple computers, and that turned out okay. So I'm willing to take the risk, and so it took that entrepreneurial spirit of Silicon Valley of taking huge risks, and for the potential of huge payoffs. I've often thought that you couldn't have founded biotechnology in Minnesota. They would go, "Oh no, it sounds risky, don't you know? That wouldn't be prudent. Oh no," And it took the business side as well as the science side coming out of the San Francisco Bay Area. And here's where Genentech started, and the city of South San Francisco, across the county line from the city of San Francisco, says the birthplace of biotechnology, and that they have about 400 companies there. When you drive the freeway south, there are multiple cranes. Every single crane is building a new biotech facility. And so this continues to be a major industry. Again, back to Herceptin. Herceptin was the first antibody that was approved by the FDA. And this is a humanized antibody, the first humanized antibody, the first antibody that was made with entirely human protein sequence. And antibodies are huge. This is the plant across the street from me. In its heyday, that one building made more money per year than Disneyland and Disney World combined. That one building made more money than the NFL or the NBA or Major League Baseball. Here in California, one year we lost the crab industry, a $5 million industry. That's one of their tanks. So one of their tanks, the value of the antibodies in one of their tanks is equal to a whole industry. And then here is a list of the top selling pharmaceutical products in the world. Yeah, you can see that the amount of money per year is staggering. On top, Keytruda, $27 billion a year. Again, that is greater than the NBA plus the NFL plus Major League Baseball. That's greater than the annual revenues of Disneyland and Disney World combined. The money is staggering. But if you look at the top 10 pharmaceutical products in the world, 8 out of 10 are made by biotech rather than traditional chemical manufacturing. And most of those are antibodies. And if you've watched any television that has commercials, even streaming services have commercials now, you've seen antibody commercials for products that are monoclonal antibodies. Their scientific names all have MAB at the end. This is a huge industry, and it's going to be getting bigger. Now, new technologies are emerging. The gene therapy, this is genetically engineering a virus to carry in DNA, to carry in genetic information, usually into children that have inherited a genetic disorder, and that will spare them from suffering. Many of these diseases have a progressive, relentless, degenerative effect. You inject 10 to the 15th, one with 15 zeros, viruses into their body. It goes through the bloodstream, finds the right tissue, invades that, but rather than invading with viral DNA, it invades with the missing gene, and that will that will compensate for the lack of that gene that they inherited from their parents. And so gene therapy, there are many, many gene therapy companies. They're all hiring. There are some in your areas. Texas is going to be huge in gene therapy. The next technology I'd like to talk about is cell therapy. The poster child for cell therapy there is the first pediatric patient to the right, Emily Whitehead. And every year she has a sign on how many years cancer-free she was. She was ready to die, and they had tried chemotherapy on her, and it seemed to knock down her leukemia, but then it came roaring back. So she was about a month away from death, and what they did is they tried this technology. They on the left, they removed the immune cells from the patient and then genetically engineer them with a derivative of HIV. That virus genetically engineers the patient's own cells to put in a new gene, an antibody gene that displays itself on the surface of the immune cell. That antibody is specific against a protein that is on top of that is on the surface of the cancer cell, and it can target it. So you genetically engineer the cells, grow up a bunch of them, ship them to the physician. The physician injects them in, and then the cancer cells go all through the body, the CAR T cells, the immune cells go all through the body, destroying all the cancer. You go from certain death to cancer-free in a month. What's the catch? Currently $500,000, but this is the future of cancer treatment. And for the first time, people are talking the C word. We're talking about curing cancer. What's coming up is now applying these technologies, growing cells to make a variety of products, to make plastics that are biodegradable, to make commodity chemicals, to make specialty chemicals, to make oils that can go into cosmetics, new oils. And you're not limited to nature. You can make new chemicals that nature hasn't seen before. And the scale that we're talking about, we're inventing a new industry that is the size of the current petroleum industry or the size of agriculture. This is the birth of a fundamentally different way to make chemicals. And let me just show you a few. Here is Bolt Threads. They make ties out of spider silk.
Jim, I'm so sorry to interrupt again.
Yeah.
Can you advance your slide? We're still not seeing them.
Oh, okay. I'm
If
sorry.
you just want to use the bottom arrow, that way we can keep up with them. I'm sorry, I didn't want to interrupt you.
I'm using the bottom arrow. Thanks for interrupting me, though. Okay. What do you see on the screen?
We're still seeing the new biotechnology, slide 8.
Oh, no. Okay. Sorry. I'm on a different computer than I usually use. So, right.
We are seeing cultivated meats now.
Okay, that's where I want to be.
Yeah, we see your cursor moving around too.
Okay, thanks.
I-
Okay.
I think you may have to actually click on each slide because the screen we're seeing is not a full slide screen.
Got it. Okay, thanks. Do you see Bolt Threads?
We do not.
Okay. Okay. Which screen do you see?
Cultivated meats still.
Okay, well, let me talk about cultivated meats. Okay, so cultivated meats are growing chicken cells in a tank. So rather than slaughtering chickens, you can take some of their stem cells and grow them in a tank and have them divide, and then collect them and make a chicken patty or chicken nuggets out of them. And the idea here is it's more environmentally friendly because you have you just will not be able to provide enough meat for the population that is coming to feed them with meat. Currently, about 1/2 of the land mass of the continental United States is devoted to meat production, and there really isn't additional land. And so if the world wants meat, you're going to have to develop a fundamentally different way of providing it, and that is going to be with cultivated meats. So we have this company, Upside Foods, and Upside Foods is doing that. The main issue with cultivated meats is affordability. The main issue is can you compete with slaughtering animals, which has been carried out for 10,000 years. Good. Let me try to advance. Can you see bacon? Okay, I'm sorry. Let me. So the other thing you can do is make protein using fungi and growing fungal cells in giant tanks and pressing those together. And fungi sort of have the texture of meat. And so it's a new way to provide meat or provide protein to people. And there are a lot of companies that are going in that. Consumer products, you may have been able to buy glowfish at Walmart. These are genetically engineered with jellyfish genes, and they are fluorescent. You put a black light on them and they glow. Or you may be able to order purple genetically engineered tomatoes. Or I have at home a petunia that has been genetically engineered with firefly genes, and my petunia plant glows in the dark. So what I'm trying to say is that this field will affect every other field, every chemical, everything that you wear, everything that you eat, everything that the fuel that you use to drive, all of that will be affected by the field of biomanufacturing and biotechnology. And I'll stop there. Sorry about the slides. I'm on a new computer, so it looked great to me. But
It's
Well, the
actually
pictures,
perfect timing.
yeah, the pictures we saw look phenomenal though. The glowfish are adorable.
So we got an interesting question from one of our attendees. They said, how real is ageism in the biotech field? I've been accepted to a master's in biotech program at UTD. Should I accept it? Thank you.
I think that this field has less ageism than most any other field. And so I often have students in my class that have gray hair and are changing careers. And so I think you have a good prospect here, probably better than any other field.
I agree. The skill sets are the same regardless of your age, right? So
Yes, and what we did is we had a project here in the Bay Area that took and especially your hands-on skill. It took people that repair aircraft, and after 9/11 they were laid off, and it shuttled them to Genentech, and Genentech loved those seasoned workers, and they worked for under a regulatory agency, regulated industry, and they just had to be regulated by the FDA rather than the FAA. So anyway, I think you have a strong prospect.
I have a question for you. As far as microbes go, what do you think the emerging trend is that people should be thinking about? Or can you study numerous microorganisms to use at the same time?
Yes, so everyone is studying a lot of different organisms. Let me give you one example here. We have a woman who did her PhD at Stanford who started a company based on her PhD research. She studied bacteria that use methane as a carbon and energy source. And she started a company right next to our sewage treatment plant. Waste methane comes out, and she channels that right into a bioreactor in order to feed the cells, and they pump out a biodegradable plastic. So there will be other organisms, but still in pharmaceuticals, we're going to stick with the key organisms that the Food and Drug Administration and other international regulatory authorities are familiar with. It's going to be for a long time E. coli, yeast, either Saccharomyces or Pichia. It's going to be CHO cells, and we'll add a couple, but it's going to be these core model organisms that you learn about in biology class. Other bacteria are going to be important. Let me make the point for the Louisiana audience that you think of Louisiana as a oil state, but it isn't an oil state, it's a chemical state. That is, the oil industry makes a variety of chemicals. Petrochemicals are in pretty much everything that you wear or everything that you put on your face, and that biotech, many of the oil companies are looking at biotech as a way to either supplement or replace the production of some of the chemicals that they do. So that's going to be important in Louisiana, especially this non-pharmaceutical biomanufacturing.
I have a question.
Go ahead, John.
Sorry. What was the name of that company that was next to the treatment plant that
Yeah,
extracted
it's
the
called
methane?
Mango Materials.
Mango.
Yeah, they just like mangoes. I don't know, but they're making a go of it. And what's interesting about that, of course, is that they have a partnership with our city, the city of Vacaville, who just gave them the land for free and want to encourage the production of biotechnology, especially the production of a environmentally friendly biotech. If you think about it, what you're doing is you're taking carbon out of the air rather than removing it from the ground. And then you're using that carbon to make your product. And so it's carbon neutral, reducing greenhouse gases. The plastic is more environmentally friendly because it's biodegradable. And so anyway, there's a public-private partnership there. And there are just companies all over the place with a target chemical that they're going to produce. You just have to have the right cell or genetically engineer the right cell, learn how to grow it in an inexpensive way, and then produce the product in a way that can compete with the production of that product that's currently going, and that's usually petrochemicals.
Interesting. Thank you.
Well, thank you so much, Jim. I do have a question. I think I'm going to email it to you and just include it in the wrap up. I just wanted to get the quick topic of what your students are doing for the algae prize competition.
Oh, okay. Well, thanks. I can do that quick. My students are, they chose an algae out of nature that makes a dye, a food coloring. And what you can do with algae, everyone thinks you shine a light on it and it gets its energy by photosynthesis. The other thing you can do with algae is you can feed it sugar, and that gives you 100 times greater cell concentration. But the minute you feed sugar now, everything else can grow in it, and you have competing bacteria. So they chose an algae that can grow at really low pH and can grow at really hot temperatures. And their strategy is to grow this extremophile in extreme conditions, feed it sugar, but now the competing organisms can't grow in it and contaminate the culture. So anyway, the idea is to make, and because it's extremophile, the blue protein that it produces is really heat stable. And so the blue protein that it produces, we're hoping, we haven't done this yet, but we're hoping you can bake with it and make blue bread. But we're carrying out a collaboration with the art department. And that's the strategy anyway.
Well, we definitely appreciate your time and your expertise, and I'm excited to see all this in April.
Good deal. Thanks, Amanda.
Thank you.
And thank you to the audience.
If anyone has any other questions for Dr. DeKloe, you can just throw them in the chat and I'll make sure to get them to him and we can provide you the answers or information later. But this brings me to our next presenter, who I'm also very fortunate to call a friend. It's Dr. Terri Quenzer, and she is a chemist and executive director of the Bioscience Workforce Development Hub at Miracosta College in Oceanside, California. And in this particular role, she leads a lot of initiatives that connect community colleges, biotech programs with industry partners and other stakeholders. And this really helps strengthen the pipeline to entry-level talent and support the workforce needs of the life science industry. And on another side of Terri's job is she also is a very big advocate for disability and accessibility in the workplace. And so she and I collaborate on that often. So Terri, you can share the screen or you can just speak, but thank you so much for attending and I will let you take it away.
Thank you so much, Amanda, for inviting me, and I'm very excited to be here. And Jim, we didn't coordinate this, but I'm very excited that what I'm going to talk about is going to give you some pathways and direction to what Jim just told you about. So I'm going to go ahead and share my screen, and hopefully I won't have trouble with advancing slides. I'm sharing my whole desktop here just because I'll jump around a little bit. But thank you for the introduction. That's all true. I did start out as my training. Actually, I started out in geology, but my PhD, my master's and PhD are in chemistry. I switched to that side. And I also, those of you who have taken chemistry courses are probably aware of being approved by the American Chemical Society. And I spent about 6 years on the committee for American Chemical Society that approves 2-year ChemTech programs. So I started on the same side as Jim, and I was in pharma for several years. So I have a whole career pathway, and if there's time, I'll show you a quick kind of how I jumped around because it's relevant too. I don't know how many of you that are not students, or even if you are students, are doing right now what you thought you were going to do with your career. So there can be a lot of changes along the way, and that's good. So now, did the slide just advance?
It did, thank you.
Okay, good. So Jim gave a really great explanation, definition of biotechnology, so I don't need to go into that. But what I would like to do is talk about what can you do with this. So as he mentioned, basically, the biotechnology fights diseases, it feeds the hungry, it addresses environmental impacts, and helps us clean things up. So I'm going to share some resources with you today because I think these will be of value to you. So this particular slide that I'm going to talk about in just a second, I'll show you where that came from. This START Engineering, it's a program within the Department of Defense, STEM, and they have a publication called Join the Biotech Revolution. Save the planet, feed the world, cure diseases, protect troops, make a difference. And this to me is one of the most exciting publications I've seen in a long time because it really captures the vibe and the energy of biotech and the really exciting things that you can do with it. And it also talks about what the other careers are, as well as, most people think of biotech as coming up with biotherapeutics, biomedical, and it's definitely a rock star of healthcare. However, there are a lot of other areas where biotech makes a huge difference or can make a huge difference and an impact. And it's for everybody. And to Amanda pointed out that we work together on projects to help individuals that have disabilities. And we're talking about a whole range of visible and invisible disabilities. And that doesn't go away. And really the bottom line for all the work we do is to build the workforce and get as many people into biotech careers as we can, because it provides affordable wages, livable wages, and in California, they're extremely expensive. So, you know, give people a chance, get people into that industry and give them career options. So that's extremely valuable. And so now back to this picture, I think out of that that publication, Join Biotechnology, is basically I don't know if you can see it very well, it's small, but I'll share the slides with Amanda so she can send them out and you can look at this closer. But I love this picture because it's very dynamic and graphic, and the things that it mentions here are, and boy, it's even small for me. You know, personalized gene-based medicine. I mean, some of the things that biotechnology can do, or what they're saying, the wonders of biotechnology, and I can't agree more. Plastic from greenhouse gases, drought-resistant crops, healthier livestock, new food sources, fuel from industrial waste. I mean, all of these things. Let's clean up what we have and work with the mess we made, and we can use biotech to do all of that. So what I'd like to do next is show you another resource, and it's a really simple website URL. So I have it on all these slides, and that is the biotechcareers.org site. And that, I'm going to jump over here for a minute so that I can show you a couple things on here before I go on into how to use it. But this site is focused on biotech careers, and there's a lot of things you can learn about, namely careers, which I'm sure a lot of you are interested in. And for the faculty here, this is something you can use with your classes. And for the students that are here, explore on this site. There's so much to see. So you can learn about careers and specific job areas. These two are both similar. You can find out about businesses. You can learn about who's doing biotech, and you can find out where it's happening, and even in your areas. So for this presentation, I'm going to focus on the careers in biotechnology, and talks about career pathways, what kind of education you need, and what the careers and job areas are. And then you can scroll down and you can explore. So there's all kinds of careers listed here. And so you can pick any of them and all of them. And if you click on them, it'll take you to a page that describes it. We just went to biomanufacturing technician downstream. So that's downstream processing. And in all of these, you're going to get this column on the left that says view a career. And so right now, what's highlighted is the biomanufacturing technician downstream. So that's what we're looking at, but you can bounce around. Let's go look at what's upstream. And in each of these cases, they give you some quick facts so you can find out what the pay is going to be, what the starting wage is, what the education's that's required for these jobs, the technical skills, the knowledge skills, the professional skills. And those professional skills are really important. That's basically how to behave in industry. And you really want to go in there with those skills, because I sit on many industry advisory committees of the community colleges in California. And I hear over and over and over again from people in industry. And I used to be in industry, and I used to hire interns myself. And my favorite ones came from the community colleges that were local to me. So the biggest thing that people in industry want are those professional skills, those soft skills. So, you know, when your professor is telling you, don't be glued to your cell phone in class or while you're working, they're not just being a parent to you. They're actually giving you some valuable advice and insight for when you get into industry. So those things are very important. Anyway, but all that information is here. You can scroll down and there's even more details about it. And these are in all of these careers. These are all links that you can go to. So I want you to be aware of that, and the site biotechcareers.org. So now I'm going to come back over here And now, bearing in mind the biotechcareers.org site, so going back to that list of what biotech can do in terms of combating diseases and extending life, we're talking about things like biotherapeutics, gene therapies, medical devices. These are different areas. And then each of these on here, when you get these slides, they're a link to these different job and career areas. And these are some examples of the job and career areas that if you learn these skills, you can use it here in fighting diseases, coming up with new medications, things like that. And the other thing is you know, when you start a career, you know, these might be jobs to start with, but if you think about it, you're really launching a career. And don't think about it as a job, think about it as a journey, and a journey through life and getting to go to exciting things. So these are one of the things I want to point out too is a few things, because you're going to notice these showing up in more than one area. So protein purification, for example, or cell culture and media and solution preparation, which you need for cell culture, quality control, bioprocessing, biomanufacturing, documentation, lab safety. These are going to show up in every area because they're needed. So these are skills you're getting in your classes that you can use in all these areas. Feed the hungry. There's a lot of really cool things going on out there. There's not only is this for agricultural industries, food and beverage industries, but there's so many new things going on with plant-based foods and with alternative meats that are grown by cell culturing. There's so much going on in the food areas. There's also a lot going on environmentally with biofuels, environmental monitoring, a lot of the same things, but you're applying a lot of these skills that you're going to need in every area of biotech. So the point of this is that these skills are transferable and you have so many options. These are such valuable skills that you're getting. And so, here we can clean energy and using less energy and cleaner energy. So again, a lot of these same skills come into play. And then, you know, also cleaning up the processes of industrial biomanufacturing, industrial manufacturing. And we can come up with a lot of clean alternatives. We're using a lot of the same skills. And so not only are there links on this presentation that I'll send to Amanda to share, but also once you get into the biotech careers site, these are all on the left side here we're in the careers, and we just went to one of the careers, and that's what got us here. And so there's so many more things you could do with this site. I could spend a whole hour or more talking about it, but explore it. I really encourage you to explore it. And then the bottom line to this is the pathways. And these pathways, basically, you can start in high school. And this, to credit Michael Fuller at Baybeck, this is an adaptation of his BART diagram, which is basically like a tube or a subway. And so you can start as early as high school or even before that, and go to your community college where you're going to get skills. And while this particular pathway shows the pathway in California, it's really the same everywhere. And so start at your local community college and get some skills, and you have so many options. You can go to work anywhere along this pathway and use those skills and hone them and get more experience. And then you can you can either get certificates and or an associate's degree. And then you have options. In California, we offer biomanufacturing bachelor's degrees at some of the community colleges, or you could go to a traditional four-year institution for your bachelor's degree. And in either respect, once you get your degree, you come out into a biotech career. But the bottom line of this is it's not like it's just that's the end game, the finish line. you're constantly, if you're a life learner, you're going to be going back and you're going to get your skills, you're going to improve your skills, and you're going to get new skills. So and these are all the things that keep you moving along in your career and moving up the ladder. And I see it happening very fast for students that go along these pathways. And you can work as much or as little as you want along the way. But I encourage you to work as much as possible and get that experience, because that's what gets you more experience. And we have an NSF Excellent grant right now. We're in our second year. In our first year to date, we've placed 26 students into internships at 8 companies across 3 regions in California. 4 community colleges so far have participated. And I meet with those students every month, and we did a Wordle about, their experience working just in the internship they did. It was 10 weeks, and the biggest term that came out of that was inspiring, but they really loved it. It was transformational, wonderful, awesome, insightful, motivating. And these are what our students are saying about that. And so anyway, with that, I think I'll hold off on talking about my career path because I think I'm out of time, and I'll just open it to questions.
Quick question, Terry. This is Amanda. With all the work you do, I know that you work across the country with different schools and industries. What does that look like for you, for the students, and for faculty?
So you mean what I do? What does what I do look like?
Well, in the sense, like, because a lot of your work is focused in the California region, but I know that you work with other industries and schools outside of California.
Correct. So my main focus has been in California as somewhat of a liaison and leader of initiatives that help provide faculty with professional development. A lot of our faculty that teach biotech have never set foot in a company when they get tagged to take over biotech. And so we try to get them as much hands-on experience and practical experience to help them work with their students. We reach out and work with people in different states as well. I am also a co-PI on the Innovate Bio, National NSF Biotech Education Center grant. So through Innovate Bio, I'm leading the initiative for DEI, which I know that's a bad word right now, but really all it means is we're casting a wide net to get as many good people trained and qualified for these jobs. So we're building the workforce, and that's the bottom line for all these companies. They need the workforce. So I don't know if I answered your question, but
I just really want students to know that even though you may be talking about California as your foundation, this is happening in states all over.
yes, oh good. Yes, and that's correct. That's true. Now, I've been told that every state needs one of me because I'm not a common participant. This isn't a common role in all the states, but it is really helpful because faculty are strapped pretty thin and stretched pretty thin. And, it takes a lot to build these industry relationships, and we try to help faculty do that. And we also bring in our own connections and really try to facilitate and make those things happen more quickly. And I'm excited that an NSF ATE proposal we submitted in October has advanced to funding consideration, and that is to build a statewide business and industry leadership team in California that can be used as a model in other states. And that business and industry leadership team will identify trends in their subspecialties and help us build skills, knowledge, and abilities standards that faculty can convert into curriculum. And that, by the way, will be shared nationally. So faculty across the country will have access to what we learn from our statewide industry advisory.
That sounds super interesting. Have you been awarded that, or is that pending?
Yeah, that's pending. We've just advanced to funding consideration. So
Well, congratulations on that.
fingers crossed. Yeah. So thank you for having me.
Thank you very much. If anyone has any questions that they think of, please just share them in the chat. I'll make sure that they are shared out, and I will be requesting all presentations to share those out with you also. So this brings us to our next presenter, Dr. Quincy Brown. Quincy Brown is an associate professor of biomedical engineering at Tulane University, and he's a member of Tulane Cancer Center's Translational Oncology Research Program. And his research focuses on the developmental and clinical translation of optical technologies aimed at improving cancer detection and treatment. And just a side note, I am very fortunate that I have known Quincy for a very long time. His wife and I grew up together K through 12. So it's a small world out here. Thank you, Quincy. I appreciate you attending.
Yeah, absolutely. Thanks, Dr. Rosenzweig. It's great to be here. I'm going to see if I can do presenter mode. Let's see if this works. Did it go to presenter mode?
It is. We see a picture, translational science and academia.
Okay. Yeah. So thanks for having me, and I really enjoyed the first two talks, and so I'm going to bring a perspective of someone who's in a research intensive academic position, but who's also passionate about translating what we do in the laboratory to the community. And so I think Amanda asked me to do a day in the life, so I'm gonna do a day, and I'm actually gonna do yesterday. I'm gonna talk about that in a little bit. But yeah, so you may have noticed that picture. You may have recognized a couple of people, and one of these is me. And so this was something that happened last August. This was a really exciting event for us. So President Biden and Dr. Joe Biden came down in August to announce some awards for the ARPA-H Precision Surgical Interventions Program. And so this is it was a nationwide competition to develop new technologies to image surgical specimens in real time in the operating room to basically help surgeons remove all of the cancer the first time. And so we were one of 9 institutes that were chosen to be a part of this. And they actually came down to New Orleans to announce it. And this is part of former President Biden's cancer moonshot. And so, yeah, this is a really exciting time, and not just for me, but for my team. And it really is sort of the culmination of 20 years of work. And so I'll kind of talk a little bit about, and we're not done, right? So this is we're just really starting to really accelerate things with this investment. But just to give a background in terms of what is this about. So, I don't need to tell you that cancer is bad. And so certainly here in Louisiana, we feel the effects. We here in New Orleans, we were in or near Cancer Alley. And so if we look at the sort of top cancers that affect the people in the US and in Louisiana, we're looking at cancers like breast, lung, prostate, colon, and rectal, as far as total incidence, and as well as mortality. And so clearly, this is early detection and better therapies, as Dr. DeKloe talked about. These things are coming along, but we need more help. And so if you think about the cancer management pipeline, so really it starts with some sort of suspicion, right? So either you take a screening test, that could be a mammography, PSA test for prostate, some sort of screen that will basically suggest that there's something that we need to have checked out. And so the next step would usually for a solid tumor would be a biopsy. And so the interventional radiologist or your specialist would then try to take a piece of that tumor, whatever it is, and then try to determine whether or not it is benign or malignant. And they would, some weeks later, you're anxiously waiting, you get this pathology report and a bill, and they tell you whether or not you have cancer. And so unfortunately, if you do, there are a few options. And so if you catch it early, for solid tumors, surgery is going to be a good frontline option. And so if not, maybe you might get hormone or chemotherapy before to maybe shrink the tumor or to make the surgical intervention more appropriate. And then possibly you could do additional therapy after the surgery if the cancer is not metastatic. If you have metastatic cancer, probably it's going to be a chemotherapy type situation or immunotherapy, as we heard earlier. And so, really early detection is great, right? So we want to detect tumors early so that we can remove them surgically. But early detection is not helpful if we don't have good surgery. And so the goal of the surgeon is to remove the entire tumor with a surrounding rim of normal tissue, and that's called the margin. And so the idea is, again, it's really simple. Surgeon is removing the tumor. You want them to remove all of it. And the way this is determined is that after the surgery, the tumor is removed, and then sometime later, another set of technologists and doctors look at the outside of that tumor and they determine whether or not there were any cells on the edge. So if there's any tumor cells on the edge of what was excised, then there's a high probability that there was cancer cells left inside the body. But the problem is that this happens a lot, right? So if we look at how often, so this is called a positive surgical margin. So if there's cancer found on the edge of the specimen, positive surgical margin means there's cancer left in the patient. How often does it happen? Well, on average, 6% for breast, which doesn't seem like a big number, but there are a lot of breast cancer cases. For prostate cancer, it can be 21% or higher. Are my slides advancing? Is everything's good? Okay. For prostate cancer, average is 21%, but it can be as high as 50%. And actually, the worse the tumor is, the more likely this is to happen, and which makes sense. So the more advanced the tumor, the more that it's growing outside of the boundary of the organ that it's in, and the harder it's going to be to completely remove. And so we really want a solution to this problem. And so what happens now? So now the tumor is removed, and they send it to a lab. And it takes about a week. And what the lab does is they take the tissue and they slice it up into slices. They call it bread loafing. They just use that analogy. It's kind of weird, but anyway, that's what they call it. And then from each of the slices of bread, they take 4 micron thick slice. They put it on a microscope slide. And that's what the pathologist, that's the doctor who looks at these, they are looking at this to determine if there's any tumor cells on the edge. So you can think of it as like an apple, right? Whereas if the excised tumor is an apple, they take big slices, and from each of those big slices, they take one very thin slice, and they put those on a bunch of microscope slides, and they look at them under the microscope, and they look at the cross section, and they try to determine, are there any cells on the edge? But what they really want to know is where's Waldo, right? So if I've got this whole tumor, and surgeon, they can't see the cancer cells with their eyes, right? So they can try to feel, but you're literally looking at the cellular level. So I want to know, where's Waldo? Where is the tumor on the surface of this, tumor cells on the surface of this specimen, if they exist? And I want to know in the operating room. But the best thing we have now is this, right? So this is looking at cross sections a week later. So there's basically very little chance they're going to find Waldo. And besides, it's too late to really solve the problem. So what we want is a technology that could look at the entire surface of the tumor while the patient is in the operating room the first time. If we could do that within 10 minutes, then we could tell the surgeon, hey, you didn't get it all, go back, take more tissue in this certain location, and then you would have a better probability of having a good surgery with a good outcome. And so that's really what this project is about. And so for the past really 20 years, I've been working on this problem with a variety of different solutions. And the one that we've been working on for about the past 12 years is it's high-speed microscopy. So we're developing fancy microscopes that can image the tumor directly. We don't have to slice it, cut it, fix it, and we just dip it in dyes. We have handlers that put it on the microscope. The microscopes are all custom, and we get these huge Google Map images of the surface of the tumor, and then we're looking for cells. Residual cancer. So what you're seeing here on the left is actually a prostate. So this is what the surface of the human prostate looks like at the microscopic level, taken immediately after it was removed. And so all these sort of squiggly things are like blood vessels and nerves, and there's fat in there. And so the idea is that, can we go and try to find residual tumor? But these images are huge. And so we want to do this in 10 minutes. So the challenge number one is image a very large area in a short amount of time at the cellular level. That's a very hard technical problem. If we're able to do that, we still then have this huge image that someone is going to have to review, and that's going to take too long. So then the second part of this is AI and ML, right? So we're developing machine learning and artificial intelligence approaches to basically automatically detect tumor cells if they're present, and then show the location of those cells on 3D representations of the tumor in real time, so that the surgeon can see, yes, there's tumor cells left, where is it? And then they have the information they need to go back and remove more. So this is a consortium. It's Tulane, but there's other universities, the local VA, LCMC, and then a startup company that came out of my lab as well. And so we call this Magic Scan. And so this is part of the team. The team is growing. So this started, we got this award last August. We've since hired, I think, 6 people here at Tulane. And we're fortunate that some of those were people who had left Louisiana, who had gone to other states, and in fact, even Europe, who weren't able to sort of get these types of jobs here in New Orleans. And then this gave them the opportunity to come back and to participate. And so just speaking about sort of the things that are happening in New Orleans, I think this is part of a bigger story of us being, more competitive in this space, and providing opportunities for folks like you to participate in this kind of research. So how did we get here? So Amanda mentioned that she went to school with my wife. I'm from Columbia, Louisiana, which is a little tiny town south of Monroe. I went to Caldwell Parish High School, and I got my bachelor's in biomedical engineering at Louisiana Tech. So that was back in 2001. And when I was finishing my bachelor's, it took me 5 years. And I didn't really know what I wanted to do. So back in 2001, there wasn't a ton of industry for biomedical engineers. And so I said, well, I'll just get a master's degree and I'll just take time to figure it out. And then I started doing research, and my advisor, so this is a much younger version of me, and my advisor there on the left, he said, hey, like, you're pretty good at this. Do you maybe you could get a PhD? And I said, no, what do they do? And he said, they do what I do. And I said, oh, that seems pretty cool. So sure, I'll do it. Why not? He said, oh, and I can pay you, so I can actually pay you to go to school. And I thought, oh, that's even better. And so that's what I did. So I stayed and I got a PhD. And I was working on glucose sensing, non-invasive glucose sensing. And so after that, it was, again, sort of a decision point. Do I go into industry? Do I go into academia? And actually applied to both. And it just turned out that, I took an interview for a postdoc position at Duke with Nimi Ramanujam, who was my postdoc mentor. And she really sold me on her vision of using light-based technologies to improve cancer management, but really focusing on impact. Like, how do we help solve problems in the clinic that can help people sooner rather than later? And so I really was taken by that. And so I was fortunate enough to work with her for 7 years at Duke. And so then I came to Tulane in 2012 as an assistant professor. And then along the way started a couple of companies. And so one of them is Instapath, which was co-founded by me and some former PhD students that came out of Tulane. And then more recently is the ARPA-H award that I sort of talked about. So just to briefly mention Instapath. So we have started a company. This company was started in 2017. I know there's some California relations here. So this is a YC company. So they were accepted into Y Combinator in 2019. They're grant and venture funded, and they're currently headquartered in Texas. But they have offices and labs in Austin and Philadelphia. And so what this company has been doing is trying to commercialize early versions of the device that we built. And they are also helping us with product development for the new things that we're building. So anyway, so Amanda said, hey, a day in the life. And so I said, I was actually working on my slides last night. I said, what would be a good day? And I was like, well, today was good. And so this is literally my schedule from yesterday. So not every day is this busy, thank goodness. But this was a good one because it covered a lot of areas. And so came to the office at 8 and started working on some stuff for class later in the day. 8:30, I had a meeting with the tech transfer office and some patent attorneys. So we had a new patent that's being allowed. And so they wanted to talk about some of the claims on that. 9 a.m., I had a Zoom call with a potential graduate student. So we're recruiting graduate students for next fall. And so I had a call with a student. After I met with her, then I had a meeting that I have every 2 weeks with a core facility that I direct downtown. It's a genomics and spatial multiomics core. And so they have a site visit happening tomorrow. And so we discussed that. And so that was at 10. 11, I had office hours with a student athlete. She was away at a track meet, and so couldn't come to regular office hours. And so met with her, did office hours. 11:30, had a weekly Zoom meeting with our collaborators at Utah and people here for a different site visit for this project that's happening on Friday. And I got there late. And so this is like you can see the pattern that's happening here. 12:30, I had a weekly meeting with my lab undergrads and lab staff. 1:30, finished that up, ate something, finished class prep, went to teach at 2. 3:30, went back to the lab, check on the grad students, anybody need anything. They built a couple of cool things. I played around with those. 4:15, back at the office, working on manuscript figures that another grad student's working on a paper, posted my slides from class to Canvas. I counted, I sent 24, this is great. I was like, cool. I was like measuring things. So I sent 24 emails yesterday, lots of Slack messages. I have 4 different workspaces. And then at 5 o'clock, go pick up my kids. Right. So that was, that's a tip, maybe it's not a typical day in their life, but it's not an atypical day in their life. So what is, so really this path is the alternative path, right? So like becoming a professor at an R1 institution, that's not the, that's the alternative path, right? That it's not there's a lot fewer of us than there are folks who go out into industry or do other things. But I think this is a great job, right? So it's a job that allows me to work with students always, and that's really keeps you young and keeps things exciting. But then I get to touch a lot of different areas. And so in academia, we kind of talk about the 3 pillars, right? So teaching, research, and service. And then I add a 4th, which is this entrepreneurship translation piece. And so just in terms of teaching, we have a reduced load. So I teach 1 class a semester, and typically 20 to 50 students per class. And those are undergrad courses. And occasionally I'll teach a grad course. And then, there's just sort of a list of sort of the things that I do for teaching. And I'm sort of going quick here, but we can always back up if there's questions. Research. So I've got meetings every week, individual lab, full lab, different meetings. We're writing manuscripts, going to conferences, grant proposals. You guys were just talking about it, NSF proposal. That's a big part of what we do, raising money to pay for the research, doing grants and project administration, communicating with agencies, doing reporting, actually finding time to read papers. And if I'm lucky, I can get to go to the lab and break things is usually what happens. Service is a big part of what we do. So this professional service, so at the university, serving on committees, firing committees, task forces. I mentioned that core facility that I work with for the scientific community. We do, journal editing, peer review for manuscripts, reviewing grant proposals. I do that 3 times a year as part of a NIH study section. And so these are just things that sort of keep us connected to our professional community. And then that 4th one is sort of this entrepreneurship part, right? Where it's working, spend a few hours a week meeting with folks at the company, helping them with their strategy, their grant proposals. And then here at Tulane, right, making sure that they're protecting the things that we build, and then trying to work with the Tulane's office to promote innovation at Tulane and in New Orleans more generally. So I think I'll stop there. And so I just want to say, like, this is, really this team of people that you see here is, it's all about them, right? So it's really about the patients and about the sort of people who get to work with me, or don't get to work with, I get to work with. I mean, that's really what it comes down to. And so I put my email up there. So if anyone is interested in opportunities, please just reach out, and happy to take questions.
Quincy, I feel like I know so much more now. This is so interesting. Does anyone have any questions? Is anyone considering looking at Tulane as a transfer pathway to a bachelor's program or a master's?
I am currently looking at Tulane, LSU, and UNO to transfer to finish my degree in biochemistry.
Well,
Great.
now you know someone, Malaya.
Great. Yeah. Happy to help in any way I can.
Malaya is one of our chemical technology students, and she is towards the You're graduating, aren't you, in May?
Yes, ma'am, I'm graduating this May.
Yeah.
I will be sending out an email too. Thank you for your presentation.
Great, thank
Thank you
you.
for coming, Malaya, and thank you, Quincy. If anyone has any other questions that may come up for Quincy or any of the other presenters, just put them in the chat and I'll make sure I get them to them. And I've also already received a lot of the presenter slides, and I'll be sure to share those as well. And then there's an iPhone user that asked, if you're going to Delgado for nursing, can you double major in biology? Absolutely. A lot of the courses align for both, but once you are focused on biology, you will be taking a few extra pathways. I'm going to put my number in the chat, my email in the chat for you, and you can email these questions to me about the majors, and I can help with the pathway plan for you. Okay, I appreciate you coming, Quincy, and presenting. Thank you very much. And this leads us right into our next presenter, Dr. Jay Shockey. Dr. Jay Shockey is a research geneticist specializing in plant genetics at the USDA. He's made significant contributions in the field for plant lipid metabolism with a focus on seed oil composition and biosynthesis. And I know that some of your research has changed over the years, but I will let you speak about that. And we appreciate you attending, as USDA is such a wonderful supporter of our Delgado programs.
Thank you, Amanda, and thank you to everyone for attending today and to the previous speakers for such interesting talks. I may have a hard time following Dr. Brown's work. We don't do anything quite as amazingly cutting edge technology as that, but I hope what I can present has its own type of interest as well. Sharing my screen now. Can you see my screen?
We can, but it's in presenter mode.
Okay.
So if you want to switch the display settings.
Yeah, I don't want to
Yeah.
be stuck with that
If you
issue
go
again.
up
Okay.
to on the top, the middle portion that says display, yep, you got it.
Yeah. Yeah. I think we're still in presenter mode, aren't we?
Yeah. So right now
Okay.
we're just seeing the
Okay. Let me give me 1 second, please. I'm going to switch the display format on my computer a little bit. Is this any better yet?
share your screen again, Jay. It's not shared
Okay.
right now. Excellent.
Okay. There we go. Sorry for the delay. Like I said, thank you everyone for attending. I want to share the next several minutes the work that we've done over the past several years, trying to answer what sounds like a fairly simple question: how do plants make oil? And trying to help you understand how a seemingly simple scientific question can become quite complex, but yet rewarding because of it. This is kind of covers some of the work that we would do in a typical day here in my laboratory at the Agricultural Research Service here in New Orleans, covering what to me is the best part about this job is that it's multidisciplinary. We're not really stuck in one particular line of work. There's kind of a series of related specialties that all combine to help us to get from point A to point B in what we're doing. It's not wanting to advance. Sorry, I'm having trouble here.
Yep, it just went ahead for us.
Okay. Before I start, I wanted to show a picture of our campus here, kind of the view that you'd get driving down Allen Toussaint Boulevard, showing the campus here at the north end of City Park. We are located on about 40 acres of federal land here in New Orleans, with a total research facility space of about 400,000 square feet. The ARS recently celebrated its 75th anniversary. That was actually in 2018, so I guess we've actually achieved 80 years now. So we've been a longstanding institution, an arm of the United States Department of Agriculture, and sort of the research branch of the USDA. We do high risk, high reward research that often is work that commercial companies don't want to do themselves because of the risk of failure, and therefore the risk of a falling share price and less dividends to their shareholders. They sort of contract a lot of that to us, and if we succeed, then they license our work, and our impact is felt in society. The Southern Regional Research Center here is made up of 6 research units, including the Commodity Utilization Research Group, which is the work unit that I'm located in. Covering the development of pilot plant research, cottonseed research, and research into sugar crops. The Commodity Utilization Research Unit is made up of 10 permanent scientists, 10 technical support staff, and 1 administrative staff. The sugarcane research crop project is funded at about $2.28 million, and the cottonseed project that I'm a part of about $2.08 million per year. The section of the project that I'm primarily responsible for is oil modification for new markets. We work in the cotton crop, and it may not be immediately clear to everyone why cotton would be a interesting research topic for the work that we do here. So a little bit of information about cotton. It's a global agronomic commodity of massive scale. Grown in about 150 countries, and provides at least some, or in some cases all, of the income for over 100 million families around the world. The overall annual economic impact is about $500 billion in US dollars. In the United States, it's grown across a wide belt of the American South and Southwest, stretching all the way from the southern parts of California around to Georgia, Florida, and the Carolinas on the eastern seaboard. And most people would be a lot more familiar with cotton as a source of the spinnable fibers that are used as the main textile that's used to produce most of our clothing, as well as sheets and pillowcases and Q-tips, cotton balls for cosmetic use, and 100 other things that are 100 other products that are found in a typical American household. We're not really interested in fiber research as much. That's being researched very thoroughly by other members of the research community. We're more interested in the seed that comes from the cotton, which is a valuable co-product of the overall cotton manufacturing process. About 20% of the value of cotton comes from the manufacturing downstream with the seed. We feel that there's still significant untapped economic potential to be exploited in development of products from the seed. And the one that I'll talk about today is the development of high oleic acid cottonseed oil. Vegetable oil in a plant, is actually the more precisely defined as triacylglycerol, the molecule shown on this slide here. This particular diagram of a triacylglycerol molecule shows the glycerol molecule on the right, the 3-carbon unit that is attached to 3 fatty acids. And each of the fatty acids here display the 3 main types of fatty acids that make up a typical vegetable oil mix in a given cell. You have saturated, monounsaturated, and polyunsaturated fatty acids, the names denoting the number of carbon-to-carbon double bonds that are present in each, with the saturated fatty acids containing none, no double bonds, the monos containing 1 double bond, and anything with 2 or more being considered a polyunsaturated. Each of these different fatty acid types, although they look quite similar to each other in a simplified diagram like this, are actually confer many different chemical and physical properties into the oil or the molecules that contain them. And so if we can control the composition of an oil in an oil seed, we can control the properties of the oil and therefore control what sorts of products we can make from it. We're primarily interested in the monounsaturated fatty acid. This particular one shown here is called oleic acid, containing a double bond between the 9th and 10th fatty acids of the 18-carbon fatty acid chain. And the reason why oleic acid is attractive is because it's heart healthy. Oils rich in oleic acid help to promote a healthy balance of high density versus low density lipoproteins in the human blood, maintaining overall cholesterol levels and keeping sort of the lipid profile of the human heart and human blood in healthy balance. So our main goal is to try and create cottonseed oils that contain increased levels of oleic acid in order to try to compete with other engineered oil seeds in this space. Why would cotton want oleic acid specifically? Why has it become a problem? Previously, more or less the entire supply of cottonseed oil that was produced domestically was used by one or two large food production companies, primarily Frito-Lay among them, to produce their potato chips and a lot of other fried food products. It was also used in a lot of salad dressings and products of that sort. But around 2005, there was the creation of a high oleic acid sunflower oil that displaced cottonseed from Frito-Lay's manufacturing processes. About 10 years after that, the Food and Drug Administration set a 2018 deadline for the elimination of all trans fat in food manufacturing in the United States, which means that we can no longer produce the shortening called Crisco, which was famous for many decades. The process that creates a liquid that converts a liquid oil into a semi-solid shortening or fat of this type required the production of a lot of trans fats in the product. And trans fats are the antithesis of oleic acid. They are very heart unhealthy. So in order to meet these standards and try to regain the market share that we had lost, there were large changes to the oil composition in cottonseed that was necessary. So the first of the main projects that we did was very much a sort of low-tech approach, requiring just traditional plant breeding processes, whereby we use the two main types of cotton that are grown in the United States. One called upland cotton, or Gossypium hirsutum for its Latin name, which is a excellent high fiber yielding variety, but whose oil contains very low levels of oleic acid. We did crosses between the hirsutum plants and Gossypium barbadense plants, which is produces much lower yields of fiber, but has some varieties within it that produce high oleic acid oils. So after several years of breeding, crossing, and analyzing of seeds and plants over multiple generations, we were an analysis by gas chromatography and other high-tech analytical approaches. We have been able to generate new cotton varieties that produce both high fiber and high seed oil oleic acid. The bar graph shown here shows sort of the stepwise progress that we've been able to achieve in doing this, starting with standard upland cotton that produces on average between 15 and 20% oleic acid. In 2020, we released a new variety called HOA1, which had shown a large increase in oleic acid, about double the amount from the starting material. And later this year, we will release a new variety, which contains a significant increase over and above that, now achieving about 55% oleic acid in the oil, or more than 3 times the amount that we started with in standard upland cotton. So are we happy with these results? Yes, this does give the cotton farmers across the American South and across the United States cotton belt some new genetics, new traits that they can breed into the lines that they use in commercial production to try to achieve both healthy oils and high quality fiber. But from a geneticist standpoint, we really didn't gain much in the understanding of the underlying genetic differences that distinguish the upland cotton from the Pima cotton that we used in this breeding set of breeding experiments. Part of the reason that it's hard to do genetics in cotton is because it has several difficulties. The genome of the cotton plant is enormous by most standards, more than 2.5 billion base pairs of DNA per cotton genome. The plants can become pretty enormous. They grow anywhere between 4 to 12 feet tall, depending on the variety, and each single plant needs an 8-gallon pot of soil. So they take up a lot of space, and they have a relatively slow life cycle. The fastest you can get a cotton plant to go from seed to seed is in about 5 months. So in order to try to learn more about what else might be controlling the oil composition and do so in a more timely fashion and a little bit easier approach, is to use a model plant organism called Arabidopsis thaliana to try to learn more about it. Arabidopsis is a much smaller plant. The picture that you see here to the left of the slide is the soil taken from a 3.5-inch square pot. So obviously much smaller than the 8 gallons that are necessary to grow a cotton plant. It's a much smaller plant. It has a short life cycle of only about 8 to 10 weeks instead of 5 months. It has a much smaller genome. There are thousands of genetic tools available to try to manipulate the genetics of Arabidopsis. And probably most importantly, I guess, is that it shares a lot of the same oil and lipid metabolism pathways with cotton and most other oilseed crops. The first little sampling of what the biochemical pathway for oil synthesis looks like is shown on the right of this slide. It's called the Kennedy pathway, which constitutes the 3 acyltransferase steps that attach each of the 3 fatty acids I showed in the structure slide earlier to the starting material called G3P. These are fatty acids that are supplied by the plastid present in all plant cells, where all fatty acids are made and then exported out for oil synthesis. However, it's as if that genetics and biochemistry wasn't complicated enough. It's actually worse than that by far. There are a lot of other overlapping pathways that produce phospholipids, membrane lipids that share a lot of metabolites with the vegetable oil synthesis pathway. You can see all of the different fluxes and different gene names along the way, each of which could play a potential role in controlling the amount and composition of the oils that each plant makes. So to really try to get at this project, to learn more about which are the key players here, which enzymes do we need to try to understand better? And really the answer is all of them. The only single component of this pathway that we feel like we really understand pretty well now is the acyl desaturation that I have boxed over to the right of the screen. That's the enzymes that control the conversion of saturated fats to monounsaturates and monounsaturates to polyunsaturates. All of the other enzymes and gene names shown in this complicated slide are incredibly understudied, and each could play a very important role in some aspect of vegetable oil biosynthesis. So how do we go about trying to address that question? What if we could knock out some of these genes, kind of start eliminating their function to try to simplify the paths that are still available to the plant to make oil? So we could begin to knock out individual steps and start to combine these steps together to create a series of mutants that will tell us more about how the flux of carbon through these pathways is controlled. Well, how do we do this? The technique that we've used is called gene editing. Some people have probably heard of it. The name that gets thrown around a lot in the popular press, probably because it is still by far the most widely used gene editing approach, is called CRISPR. It's an acronym of this terribly long name that I won't try to reproduce for you. CRISPR is good enough. The reason CRISPR is powerful is because it's been made into a very easily manipulatable 2-component design that even small labs can take advantage of. The 2-component system requires a gene encoding a DNA nuclease enzyme called Cas9 and another molecule called the guide RNA, which, as its name would imply, is a small RNA molecule that finds its match in the genome of your plant of interest And that alignment of the guide to the target site in your gene of interest attracts the Cas9 enzyme, which settles around the structure shown here and acts as a pair of double scissors, cutting each of the two strands present in the DNA at the target site. DNA breakages are a dangerous thing to happen in a living cell. So plant cells and every other type of cell in nature will quickly try to repair the cuts that the Cas9 enzyme have made. But in the process of doing so, the cells make a lot of mistakes. Extra bases get inserted, some bases get left out. And so you're creating mutations, by taking advantage of the cell's sloppy DNA repair, we can make mutations in the gene of interest. So a typical day, or I guess I should say day, week, month, year in my lab, is sort of repeated rounds of identifying new genes that we're interested in trying to knock out in our plant lines using the CRISPR technique, building the DNA plasmids that will carry the two-component system that we need to target the gene that we're interested in, transforming the plants so that the Cas9 and guide RNA are expressed inside the cells of that plant, then growing out multiple generations of those plants and analyzing both their seed lipids and their DNA to try to identify what we would consider to be finished lines that are fully edited at the gene of interest. We've completely mutated all copies of this gene in the genome throughout the plant so that now there will be a loss of function for the enzyme that would have come from that plant. And we can now begin to look at these lines to study changes in their composition or content of lipids and to look at other traits that are present in them. So I'm maybe a little behind time. I'll skip past this slide. This just shows don't worry about what the names mean. These are just names of particular genes that we've knocked out along the way. We start out making single mutants, convert them into double mutants. We start out with triple mutants that we convert into quadruple mutants. And then those get converted even as so far into a line that gets missing 5 different genes in the pathway that I showed. This gives us a wide range of genetic types that we can use to study the production of the oils. What's important to note here is that none of these genes directly control the saturation state of the fatty acids I talked about. We're not interfering with the plant's ability to convert a saturate into oleic acid or convert oleic acid into something else. We're just controlling the enzymes that control the flux of all of the oils through different lipid pools on their way to the finished product. So these stacked CRISPR lines have achieved several different step changes in the production of oleic acid, starting from about 15% in the normal plant up to the mid-30s with a couple of our mutants to 47% in a really interesting double mutant, all the way up to about 63% in one of the lines that's missing 4 genes and our plant that's missing 5 genes. So we've again created about a 4-fold increase in the amount of oleic acid that was present in the seeds of normal Arabidopsis plants, all the way up to the ones now that we're presently studying. So one of the things that can come from these types of mutational studies is that there are additional traits that you didn't see coming. And so we always have to prepare to look at all aspects of the plant's growth to make sure that the changes we've made in the oil composition hasn't also affected its ability to grow normally. And if you look at some of the plants we show here, controls on the top, comparing the different mutants on the bottom, you can have relatively complex mutants lacking 3 genes, as in this case, or 4 genes, as in this case, that are still almost indistinguishable from the control plants. However, anytime we noticed that anytime we started to include a mutation, this gene called FAE1, we started to have problems with the seed's ability to germinate and establish on soil. This quadruple mutant is particularly bad. But what was interesting is that when we knocked out the gene called NPC6 in this 4 mutant line that I just showed in the previous slide, we were actually able to restore some of the seed's ability to grow normally. So this was an unexpected set of findings that gives us a lot more to dig into. So to conclude, from a professional outlook standpoint, a typical day in my lab incorporates several different scientific disciplines, including chemistry, biochemistry, and molecular genetics. We try to plan each day carefully amongst the team so that each person is tasked with specific goals and objectives that can help to get the most out of each 8 to 5 day that we have. And every day will include multiple successes and failures, and which I would encourage all of the students listening today that are envisioning a future in science to be prepared for a lot of both. There is a lot of failure that comes along with this, and you can't be afraid of that, because both the successes and the failures help us to learn more about the underlying science that we're studying. Scientifically, we know that CRISPR is a powerful tool, and that's been verified from our work. We've also verified kind of what we already knew, that many genes and overlapping pathways control the content and composition of seed lipids in plants and other organisms. And that some of the genes, despite all of the work that we've continued to do, there are still many genes in every organism whose function remains an unknown. And even when combined with other mutations that should help to reveal those roles, sometimes they still remain hidden. And that other genes actually play multifaceted roles that we didn't see coming. Ones starting with the things that we thought we would see, and then sometimes overlapping into areas that we didn't. But we welcome these opportunities as well, because, as I said, it gives us new research angles that we didn't even know were going to happen. I'm going to finish today's slide by acknowledging the staff at my talk by acknowledging everyone at SRRC, all of the assistants that have helped, including Mr. David Allen, a recent graduate from Delgado Community College, who's actually done a lot of the CRISPR analysis in Arabidopsis for me and has been an excellent addition to the lab. Also would like to thank Cotton Incorporated, USDA, and National Science Foundation for their financial support that's helped to fund some of this work. And thank you for listening, and I'd be happy to take any questions.
I just have a quick question, Dr. Shockey. Are you
Yes.
Looking for any interns in the next few months?
We're always looking for new talent. I guess we do have a lot of existing summer intern programs. We have partnered extensively with the ORISE program, which is how we essentially 3 of the people shown in this slide were all brought on board in one form or another through cooperations with ORISE. Like all other federal agencies, we are going through a little bit of a challenge right now with some of the ongoing efforts to downsize the government. So there is a bit of more uncertainty in place now than we would have ordinarily. But I hope and expect that we will be able to weather the changes that we're going through right now, and reestablish our normal operations sooner rather than later. And certainly would encourage any of the students that are interested to reach out to me or to anyone else here at SRRC to inquire about the interning possibilities and maybe other future job options as well.
Well, thank you so much for your time and your expertise. And if anyone has any questions, please just leave them in the chat, and I will make sure that they get to Dr. Shockey. And as he said, a recent graduate from Delgado is now working with him. So USDA is a great supporter of our students, our research. So please don't hesitate to reach out to any of us to help support your goals. So thank you again.
Thank you.
Our next speaker is Miss Jessica Kong. Did I pronounce that last name right? Okay. And she is the Senior Manager for Market Development at QIAGEN. And she's going to focus on investigative genetic genealogy. And she's also a very active individual in missing person and cold case advocacy. So thank you so much for taking the time to attend today. And I will let you take it away.
Thank you, Amanda, and thank you, Charlie, for allowing me to speak today. It's such a pleasure and honor. Just quick technology check, is my slide showing? Okay, so I'm going to put it in presentation mode. Let me know if anything changes, goes away. And I can't see the screen anymore. Let me know if it's all good now?
To us.
I'm sorry?
I said it looks good to us.
Perfect. Perfect. Well, like I said, really excited to be here with you all today. I think there's 30 something of you in the audience. I I do work for a biotech company, so I think that's probably of interest to some of you. One thing I did want to clarify, some very flattering mistake. I do not have a doctoral degree, so I'm not Dr. Jessica Kuhn, but I do have a master's of biology and science, which is kind of an MBA degree designed for people that are interested in entering the biotech field. So I think with those two pieces of information, my session today will be informative and interesting to you. So a little bit of what we'll cover today. Just a quick agenda. My name is Jessica Kuhn. Like Amanda said, I work for a company called QIAGEN, which I'll tell you a little bit about in a minute. It's a biotech company. And then we'll spend the rest of the time talking about what is forensic investigative genetic genealogy and how does law enforcement use it. And then at the core of it, there is a website called JetMatch, which is near and dear to my heart. We are the steward of JetMatch. We'll talk about how that is used by the general public, law enforcement, or forensic community. And why should the general public care? Why should people that are interested in biotechnology care, and how you can be involved and how you can help? So with that, if I could take a minute to tell you about QIAGEN as a biotech company. So $2.1 billion company. It's been around for, I think, 30 plus years. And even at this number, it's apparently considered a mid-sized company. So we're not one of the biggest players in there, but we've been around for a long time. And so if any of you have worked in a lab, have walked into a lab, you will not have a hard time finding a QIAGEN product in the lab somewhere. And we have a whole suite of different products, services that we provide to people that work in research, all types of research. But the division that I belong to is one called HID in forensics. And I myself have worked in have done marketing for life science companies my entire career. And jokingly, I've recently told people for the last 5 years I've been working for a biotech company in the death science, as a contrast to life science. But that's what forensics is. And the majority of our customers are in the government sector, as you can imagine. Most of these labs are kind of affiliated with law enforcement or public safety or criminal justice institutions. So that is what QIAGEN is. It's based in Germany. The America headquarters, though, is in Germantown, Maryland. And I always say that's maybe the way of Germans trying to be funny, right? Headquartered in US, call it Germantown. But I'm in San Diego, California. So I'm a remote employee. And like I said, I do marketing, all kinds of different marketing. So on that note, a little more about, my role, like Amanda said, when it comes to cold cases, missing persons. Obviously, I am not law enforcement. I don't have a background in law enforcement, but with the sort of taking on the role of explaining new DNA technologies to law enforcement, I've had such a kind of I pinch myself every day on what I get to do for a job and the people that I work with. It's hugely inspiring. It's awe-inspiring, and it's sometimes, this is a segment of our society that looks at things that most people either don't want to look at or don't have the wherewithal to process, to understand. So so whether it be, homicide, sexual assault, missing persons, cold cases, and, the people that work tirelessly to solve these cases, they are amazing. So this is what I do, working at a biotech company that have forensic offerings, and then explaining the new DNA technologies to law enforcement to help them, expand their toolbox when it comes to investigations. I also belong to an organization called the International Homicide Investigator Association. I am on the board of directors with them. I help them with various things, some marketing, some other kind of responsibilities. So feel free to Google them. Great nonprofit. Lots to learn from them. So I'm here to talk to you about what forensic investigative genetic genealogy is. The acronym for it is FIGG, or I sometimes call it FIG, just because it's a lot easier to say. So I have a series of pictures to show you to kind of to jog your memory, and you must have seen it online at some point in the last 6 years. The forensic investigative genetic genealogy has been kind of been a the I sometimes hesitate to say it, but it media loves using it because it really truly is amazing when it comes to its investigative power. But the cases, there's a lot of kind of high profile cases that it's been associated with. So I am sure you will not have completely find it completely foreign. So there's a series of like these high profile cases, and some are active ongoing cases. So the court system will determine if truly they are guilty of the crimes that they've been accused of. But these are, some of the high level or high profile cases that you may have heard of: Joseph D'Angelo, aka the Golden State Killer case. And this was John Wayne Gacy, and so FIGG was used to ID one of his victims a few years ago. And this guy is by the name of Brian Koberger. This is a case in Idaho that there's a lot of media attention on the case. And this is a case up in Suffolk County, New York, a very just heinous series of crimes that this guy has been accused of. So these are to serve the purpose of kind of jogging your memory. Maybe this is where you have heard about forensic investigative genetic genealogy. But there are some other types of investigations that FIGG has been a part of, are these sort of historic either missing persons or mass graves, or just recent disasters like the Maui fire, where the bottom line is FIGG is used in human identification. So whether that be a offender of crime, of the particular types of crime, or remains or unidentified persons that need identification, FIGG could be used for. So how does it work? How does FIGG work from what we call from DNA to ID? So when it comes to investigations, whether you start with a person that is unidentifiable, usually a deceased person, not all the time, or unknown crime scene DNA, the goal is to identify who this biological sample belonged to, right? So what you start with, for example, is crime scene DNA, unknown crime scene DNA. What you need to do is sequence that piece of DNA. And this is where kind of the rubber meets the road, right? In the forensic space, a lot of times crime scene DNA are either in very minute amount, or they've been exposed to the elements, so they are very degraded. So this is where in maybe some of the other presentations, I don't know if you guys have heard about DNA sequencing, but a lot of times in the clinical space, you have like a whole tube of saliva or, fresh blood samples. Very different in the forensic world. And this is where things get really tricky. So you sequence that DNA, and at the end you get something, what we call the SNP profile. SNP stands for single nucleotide polymorphism. For biotech students, that's probably not super foreign to you. But once you have that SNP profile, what you would do is upload it to a website like GEDmatch, which we'll talk about what GEDmatch is, how it works in a little bit. But what just in short, it is a genealogy website, genetic genealogy website. And there are lots of rules around what you can have access to, what kind of cases qualify for this type of upload, and we'll talk about that in a little bit. But once you upload a GEDmatch, you get back what you call a match list, or a list of folks that this unknown profile share genetic associations with. And there begins the work of a genealogist that does what we call the genealogy research, or building family trees. Basically, with this list of matches, or people that share a varying level of genetic association with your unknown, how do they relate to each other? How do they relate to, and this in turn will inform you on how these people are related to your unknown sample. So eventually, the result will inform investigators on how to proceed with their investigation, whether they need to reach out to the community for more what we call reference testing to expand the family tree, or sometimes in luckier scenarios, they'll get handed a list of sort of investigative leads to further investigate. So that is kind of how it works in a nutshell. How does QIAGEN play into any of this? I wanted to show you a little bit in the QIAGEN HID world, the types of products that we service. These are all biotechnology products that in the QIAGEN HID portfolio. So starting with samples, I won't go into like what these all are and what exactly they do, but they all revolve around
So first
DNA
question.
analysis. Yes, ma'am. Amanda, did you say something?
No, I think
I think
someone
that
just
was an
accidentally
accident.
came off mute.
Okay, I'll keep going then. So revolving around DNA, starting with evidence, when you have the actual DNA, once it's purified, quantified, sometimes amplified, then you can do all sorts of analysis and at the end report on the identity of who this DNA belongs to. And so there's all kinds of products, software, hardware, consumables that fall into our group of our forensic product portfolio. But the two that are most relevant in the FIGG world are what we called a DNA sequencer. In this case, it's the MySeq FGX, and then that website JetMatch that I've mentioned previously, the the genealogy website. So these two kind of product offering is what makes us so interested in FIGG, not only with law enforcement utilizing it more to solve cases, but also educating the public on how they should think about FIGG and if they are willing and comfortable with how to participate, which I'll tell you about in a moment. So just a moment to clarify a few common misconceptions. I call it pop quiz, but you're not being quizzed. You probably have heard about companies like 23andMe or Ancestry DNA. These are the two of the largest, what we call consumer DNA testing companies. So you spit in a tube, your saliva to them. They give you results on a lot of, some are more healthcare related, others are more lifestyle related. And companies like Ancestry, it informs you on who you're related to, right? These companies don't allow law enforcement searches. So one of the biggest misconception is people that have taken 23andMe tests or Ancestry tests, they think, well, I'm in these like DNA databases somewhere, right? Like, yeah, law enforcement can use it. And these are the, for people that are kind of supportive of this type of utility. And that is actually incorrect. If you've never taken the step of downloading your profile from Ancestry or 23andMe or any number of, there's like 20 something such companies that do similar kind of services. If you haven't taken the time to manually download your profile from these companies, creating a profile on JetMatch, uploading your DNA profile to JetMatch, law enforcement cannot use your profile. So for some of you, maybe that's comforting to know because you don't want anything to do with law enforcement investigations. For the others that are like, hey, I haven't done anything wrong in my life, I'm comfortable, like, if law enforcement can use this technology to solve serial killer cases, why not? What you could do is download your profile, upload a JetMatch. I make it sound really simple. It actually isn't. But if you're interested in doing that, feel free to reach out to me and I could walk you through how to do that. Or there's no shortage of online material on how to do that you could find. So just a kind of moment to clarify things. So like I said, JetMatch is a genealogy website. And what happens is people that have taken, and JetMatch does not issue DNA tests, right? So that's a big big thing that makes JetMatch different than a lot of the other companies. So what most people do is they have taken a DNA test from some other company. And one thing, let's say you took the Ancestry DNA test and you're looking for, maybe you're adopted and you're looking for your biological family. So let's say your biological family is doing that and they took 23andMe test. right? You're not going to find the connection between an ancestry test and a 23andMe test unless both of you do the sort of manual work to upload your profile into GenMatch. So what GenMatch is a cross-brand, cross-company repository of testing or test results that the purpose is for people to find genealogical connections, find family members. And so what we also have is a portal called GenMatch Pro. That is for the part of the website that law enforcement can use. So I wanted to spend a little bit of time telling you how that is all done, because I think for people that are interested but maybe are a little apprehensive, they're like, well, how is law enforcement using this? What kind of rules and regulation do they have? And like, is it like all bets are off and they just look at anything? So no, the answer is no, right? And all the profiles that law enforcement has access to are profiles that have people that belong to people that have consented to such utility. So, like I said, GenMatch is the public-facing sort of consumer genealogy website. This graphic needs to be updated. We have over 2 million users or profiles as of last year. And the portal that law enforcement or forensic professionals can access is called GenMatch Pro. So there is a piece of document called the Department of Justice interim policy that sort of dictates what kind of cases forensic investigative genetic genealogy can be used for. So cases that qualify for that under those guidelines can be uploaded to GenMatch Pro. And what through GenMatch Pro, the forensic or law enforcement professionals have a special set of a different number of tools that they have access to. It's the same type of genealogy workup that people in GenMatch do. So on that level, there's not really any difference. One thing I will say that is different in GenMatch Pro is there's more sort of security features, because these cases are, active investigation. So people in GenMatch cannot see what the sort of within the cases of these individual kind of forensic investigations, what's going on with them. So that's kind of a nutshell on how GenMatch and GenMatch Pro are different. So I mentioned the DOJ interim policy on what investigators can use FIG for. So these are kind of a brief list of the types of investigation that investigators can use FIG for: identification of perpetrator of violent crime. Are those some of the criminal cases that showed you earlier in the presentation. Disaster victim identification. As human beings, we are very good at conducting atrocities toward each other, very unfortunately. So there is very much the need in the forensic world for identification of, a lot of times, masses of people. And then again, in the same vein, unidentified human remains, decedent identification, and then on the more rare occasion, living doe identifications, people that have turned up in places that, under whatever circumstances, don't remember who they are. But of course, they have to be involved in some kind of criminal investigation before FIGG is utilized. So this is just more data that, if you're curious, that will inform you on, does FIGG really work based on our population? There's a researcher by the name of, at one point he was I think the chief scientific officer of MyHeritage, another consumer DNA company. He did a research and showed that, given the population size, how many people you have to have in a database that will allow you to identify what proportion of the population up to what level. So it's feel free to Google that. It's really interesting, and just in the interest of time, I won't say too much about it. There's also a website that tracks the number of cases that have been solved by FIGG. As of the beginning of last year, I think there's already over 1,000 cases that's been solved since the what we call the watershed case, the Golden State Killer case, that came on the radar of the public in 2018 or 19. So it does work. It has worked. It works very well. So the next part is, how can you help? What do you do? If you've taken a DNA test, let's say Ancestry or 23andMe, I would encourage you to think about joining GETmatch. This is a campaign that we call the Genetic Witness Program, and really it's just the name that we give into this kind of activity that millions of other people have done, which is being part of GETmatch. And in the GETmatch, once you uploaded your profile into GETmatch, there's something called opting in and opting out. If you opt in, your profile will only be used for the purpose of identifying unidentified human remains or decedents. If you I'm sorry, did I say opt in or opt out? If you opt out, your profile will only show up in the investigation of unidentified human remains. If you opt in, that's where you allow your profile to show up in the searches of law enforcement identifying offenders of serious crimes. So I encourage you to look into it. There's a lot of online information that you could look up to find out what this is all about. What are the cases that have been solved? There is, like I said, over 1,000 cases, and that's just the ones that have kind of media mentions. Or if you have questions, feel free to reach out to me. This concludes my presentation, which granted is very, not a lot of time to go into detail with any of these things, but feel free to find me on LinkedIn. I post about fake a lot, what law enforcement is doing, just kind of the interesting aspects. And thank you again for, Charlie, your invitation, and the work that you do in the missing persons world is just tremendous. And I am so honored to be able to share a little bit with you today about fake, and open for questions.
Thank you, Jessica. A lot of my forensic students are here, so I can't wait to see what they think about it.
So I
Jessica,
know we don't have a ton of time.
yeah, just due to time, I do have a question to ask, and it may be something that's better to follow up with. Has any of this been used with some of the high-profile animal cruelty cases?
That is a good question. I am going to guess the answer is no, because the DOJ interim policy, like I said, that it can only be used for identifying human remains or criminal offenders, but it's only for violent crimes, and there's a FBI definition of what violent crimes are, and there's homicide, sexual assault, but I have to imagine they're all defined for crimes against humans. So, but I don't know for sure. That's my 2 cents.
Thank you so much. This was so interesting and lots of great information. And not only is important for Charlie's forensic students, but also for all the Chemtech, Biotech students with the skill sets that they are learning. So thank you. Appreciate everyone's patience. As we move forward, we have our next speaker, Dr. Egan Rowe. He's the algae manager at the Thad Cochran Marine Aquaculture Center in Ocean Springs, Mississippi. And in this role, he oversees the cultivation and management of algae species used in marine aquaculture research and development. And he has a lot of other titles with his name, but this was probably the one that focused most on what we discussed. So I will let him take it away. Appreciate you attending. And you can share your screen if you want to. Egan, it's up to you. He was, yeah, there he is. He is on here. Egan, do you need...
I'm on here. Sorry. Right as I pushed one button, it basically created a new desktop and threw everything sideways. So I'm going to reshuffle the deck real quick and see if I can get this to share the screen, and we'll go from there. Can you guys hear me okay? Okay. Let's see. just. All right. Are you guys able to see this?
Not yet.
Okay. Try another one real quick. Sorry about this. I did try to troubleshoot this beforehand and it how about now?
You can see your screen. And
With no
it's
slide?
just, it's not in presenter mode, but that's up to you.
Let me see if I can just, you know what, we're gonna probably have to go that route. Okay, can you see the slides now?
No, now they disappeared.
Okay.
They're back.
Okay, all right, I think we got it. Can you see them change now?
As you click on them, we can.
Okay, I'll take what I can get at this point. All right, so thanks Amanda for including me in this. This is definitely right up our alley, and I just wanted to let everybody know, I manage the algae program at the Marine Aquaculture Center here in Ocean Springs, Mississippi, with the University of Southern Mississippi. There's numerous titles. There's numerous. It's actually at the Gulf Coast Research Lab is our actual campus. So I think I've covered everything there. I do want to correct, my degree is a Master's of Marine Science. So I don't have my doctorate. I finished my graduate work and then went right into industry, but I'll kind of go right through this real quick. I did kind of shuffle the deck on this presentation too for to kind of follow your outline of the day in the life. And so with that, I did want to kind of just specify that my the daily operations for us, and as you go through, I kind of organized it so it is kind of our operational workflow, so everybody can kind of get a feel of what the day would go like as we go through the process. So we usually kick off early at the center, and that's usually around 7 a.m. since we have to really have algal feed out by 10 for all the other end users at the facility. So I'll describe that a little bit more, and that includes AM checks with the systems, harvesting the systems, and then routine measurements. So it's kind of a busy morning for us. Once we get on the other side of 10 a.m., usually then things start to open up and then we start to move into analytical work. So the late morning into the afternoon is data processing, coordinating operations, and then conducting more advanced analytical work. And then the end of the day really is, finishing, restarting other systems, completing kind of the advanced, instrumentation analytical work, and then coordinating operations for the next day. So it's one of those things where the day is a pretty routine day, and we tried to build in a lot of ability to kind of adapt to curveballs. But it's, when you talk about a fast-paced work environment, we are definitely that, and it really kind of involves really good team skills, and that they coordinate things really well, and they're able to work, to kind of anticipate and overcome, those daily curveballs that are thrown. So with that, I'll just go into my background. Generally speaking, I started in blue water oceanography. I was interested in phycology or algal cultivation relative to zooxanthellae and coral ecology. Joined marine science program, and that took me more into population dynamics or planktonic population dynamics of bacterial plankton and phytoplankton, as well as developing skills in maintaining phytoplankton cultures. That evolved into what is my phycological and then applied algae biotechnology background, which then evolved into biofuels, but it really began through a big third wave of biofuels development that started with a lot of strain high throughput screening of a lot of different strains that ultimately build into strain development research. And then we had a pilot facility that was built in Hawaii that once that facility was complete, the consortium, I moved over there and we started to implement things. So I've had the fortune of what I say working in theory land at the benchtop level, but then carrying that all the way forward in applied science to where you're able to basically develop that at a commercial scale and see, you know, how to optimize those systems and how they actually, each species that you're developing is capable of performing at different levels. So then lastly, after kind of leaving biofuels, which is biotech heavy, I moved into aquaculture and more specifically live feeds production, which that's what I'm currently involved with now at the Marine Aquaculture Center. So I think that gives you my background just in a nutshell. So here at the Marine Aqua, or at TC Mac, I'll just kind of go over that. We're basically centrally located, and I'll just say the Gulf at this point and call it at that. And we basically, what aquaculture means is we are specifically focused on marine aquaculture. So that's obviously marine species, but basically cultivating, rearing those organisms, both for commercial, private, public sector projects, what have you, and really alleviating those bottlenecks. You can kind of see our mission statement here. So that's basically what we're here to do, is really work with a variety of partners to kind of find out where technologies need to be advanced and be there to provide that, you know, that platform to do so. The alga program specifically, we include both micro and macro algae, so both phytoplankton and seaweed. That microalgae program is what I'm going to focus on most for this talk, although I will preface that we do have our macroalgae program, and that's more being geared towards IMTA, so an integrated multi-trophic aquaculture project that is going to be an offshore project that's beginning this year that we're working with NOAA, University of New Hampshire. So that one we're excited to be have been excited to develop those technologies. I will also preface that what makes us really unique in marine aquaculture is that we use RAS systems. So those are recirculating aquaculture systems. So what we do here could technically be done anywhere on land. You could move it to Ohio or Iowa. And that's the other side of that, is that we're biosecure in the sense that it's more advanced systems, but we're able to do things in a manner that is biosecure, and those systems can be replicated onshore. So it gives you a lot more stability in the performance of your systems as well. The algae program beyond that, just a general background in macro or microalgae culturing and mass production. It really is fundamental, since these guys are primary producers. You know, they're unicellular organisms and they're base of the food chain. But this really came about with advancement in microscopy. And then the shellfish industry was really able to take that further, was the first industry to really start commercializing algal cultivation purposes. And then in the 70s, biofuels came along, and that's basically when the price of oil goes up, there tends to be a big investment in algal cultivation for biofuels. I was involved with the third wave of that, with and most specifically Shell Oil's investment in that venture. And then microalgae now is being seen as another really on the cusp of a lot of, especially offshore algal aquaculture and mariculture endeavors. So that's one we're looking to be involved with ourselves here this year. More. Then I guess to begin with, this is just kind of a general overview of how microalgae cultivation goes. And it really starts with just small scale culturing. You scale it up through your seed train, and then you reach a mass production level. So I'm going to go through that process, and I just like to kind of specify here that there are three main types of algal production. There's batch production, continuous production, and semi-continuous production. They all have their strengths and I would say some weaknesses or advantages. It really depends on what you're doing with them to see which would work best. Just to kind of simply state it for those that aren't familiar with it, is batch production is like a batch of whiskey. You make it, you let it age for a period, and then you harvest it completely. Continuous is an instance where you're basically continually coming in with your medium, if you will, and you're diluting out your production amount at a constant rate, and that allows you to produce continuously, and that has some advantages. And then semi-continuous is blending the two methods. So you're operating a system semi-continuously in that you're harvesting it at a continuous, an interval, usually days, and it's restarting itself. I tend to use that a lot more because it provides some advantages, and it also, from my background, it allows you to manipulate the physiology of the culture and do some more advanced things, so you can take advantage of that method in a variety of ways. So as I go on, our day kind of starts off, the first thing in most microalgae places is you've got to do your small scale culturing work or your open transfers in a clean environment first thing, and then you kind of move on through your day. So that's what's being done in this picture here, is just your clean transfers, and that really involves people that have good laboratory cell culturing skills is something that we really sought after, and you got to execute. Since if you don't execute, this is really the stage that can cost you a lot in contamination and other problems building up downstream. It also, I like to point out for those that aren't as familiar with algal cultivation, this is also where I use to really force the growth rate. So I kind of use the expression, this is kind of where the gas pedal is. This is where you set the growth rate in your whole seed system. So if it's lagging or if it's on the other side, you'll see it downstream immediately. And if you get this right, it really makes things go smoothly downstream, or I should say upstream, I guess. Then, or downstream, I was right. Okay, so then right here, just to give you an example, is our maintenance cultures. And this is basically where the working stocks, I just kind of like to use this as an image to kind of show you what we do. You'll see a different colorations, that's species specific. And then the color density is really just showing you, or the color gradient is showing you density. So that bottom shelf has darker cultures. And this is really where we are showing you how we're forcing those growth rates. The lighter cultures are at the top moving down, and then we dilute them. And so on. The other key thing about having the different species that we have is both size range and then nutritional composition of different species. So some species are smaller, which can be fed to different end users at different stages of life. And then as the cultures are bigger, larger species can be fed to more size specific at larger ranges. And then the diversity of species, in a lot of instances, certain species have different nutritional compositions, as well as how you're growing the culture can dictate what that nutritional composition is at the harvest stage. The key things with algal cultivation, I would say right here, light, temperature, the carbon source, nutrients, and then the water quality itself. If you can meet all those metrics, usually once you get to density, it's going to be your light, it's going to be your limiting factor. But contamination is probably your biggest risk at a facility. You're constantly making sure that you're executing properly with clean techniques, sterile transfers, such so that your culture isn't open to major contaminations. And here at the Aquaculture Center, it's even more of a risk because you could have a healthy culture that has a contaminant in it that doesn't, a microbe, a bacterium that doesn't have any effect on the culture itself's productivity. In a lot of instances, I say, you're looking at a good, healthy microbiome kind of stabilizing your culture. But if you're building up a pathogen, such as for us, Vibrio or Pseudomonas, that is a threat to your end user. So oysters for us or some other finfish research project, that if we're carrying that through our feedstock, then that's a problem for them. So that's what we're always screening for and working to alleviate. But that's just an example of kind of the factors you're dealing with algal cultivation. The next thing is the growth curve. I just point this out in that with algal cultivation, especially through the seed train and your scale up process, it's really key that you're executing and keeping things in the exponential phase. If you get into your really stationary phase, you can risk, real stability in your production operation, as well as possibly, your production being collapsing, or your culture's lagging significantly. So the scale up seed train I've kind of just depicted here is just gives you kind of an example of that. And that is really the process we use to go from like a 1 liter flask all the way up to, a 100-liter vessel. And you're usually scaling up, if you can, at a 1-to-10 ratio through your whole process. And I think right here, I kind of have an example of what we do here in house. This is specific to our facility, but it is representative of, most facilities. You're going to start with the working stocks that aren't on aeration. Then you're going to move to aeration, which that's allowing you to put air in, and it's also allowing CO2 mix, which it allows you to both get higher density, both since you have a carbon source that's at a higher rate, as well as the mixing of air allows you to get better light penetration to the culture. And then we scale up to our photobioreactor systems here, all the way up to 1,000-liter system that we use here at this facility. Mid-scale, I always kind of specify this when I give these talks, is that you can use mid-scale both as part of your scale-up scheme, and also it can be a production level. We use it a lot here to supplement production with different speeds, especially for research projects. But it often is used as an experimental stage to kind of test bed your production at large scale. I will specify, just for my career, is that if you're using mid-scale experimentally, it has to be a good representative of your large scale stage. If it isn't as good of a representative, you're not going to carry that forward. And so all that data that you're building on can be for naught if it's not a good representative of your large scale system, for instance. This is an example here where we use a semi-continuous production, which this is being diluted here on the right on basically a 2-day grow out, I think is what we've been doing with that system. And then these other cultures, like I said, are being used batch production. And those are growing out. As you can see, they gain in density. The darker bags are going to be harvested. And then the semi-continuous, that's being diluted and basically restarted with the diluent that's left over on a 2 to 4 day cycle normally here at our facility. Then we ultimately get to this large scale stage. I do couch that this is large scale at our facility. In earlier parts of my career, this would have just been another scale up stage. This is what we use to produce, and that just kind of gives you a gauge of what you're doing is specific to the operation that you're supplying feed to. And most commercial large scale production, they're going to be at, hundreds of thousands, if not millions of liters of for outdoor raceways, if you will. The other end here, I do want to specify for us, this is kind of part of our routine, part of our day, is the front end is we are doing the routine measurements. A lot of more advanced systems allow you to do that in situ within the system in an automated fashion, but the upscale stages and the smaller vessels, that isn't as feasible. So we spend some time with our daily checks on the top end. That's our routine monitoring. The more advanced analytical stuff and measurements we do is the lower end of this. So the biomass and biometric measurements, nutrient uptake, and advanced methods with flow cytometry, CNN analysis. Those are the latter part of the day or for research purposes currently. I do put this one here because this is an example of where we worked on this prototype system on the right that we're currently using and planning for our new facility. And it just shows, this is an instance where the design of the system allowed, a bigger increase, and it's basically light penetration. The system on the right we developed since we have 2 banks of light. The one on the front of that is shut off, but the other system is more round. It doesn't have the light penetration. So that one on the right has close to, 18% production or productivity compared to the other one. So it really worked out as a real space-wise as well as productivity improvement for us. And that's the system on the right we're looking to expand for our new facility that will be coming. For photobioreactor systems, that's what I've kind of been showing for the most part. Those are what we currently use for our production stages. And that just means a photobioreactor is a growth vessel system that has light automation as well as other automated, in our instances, usually that's CO2 input, but our systems also have some nutrient fluid controls. So it really depends on what you're doing, and you can get really advanced with production systems. And I would say that's one thing in my career. It is very multidisciplinary, and I've used a lot of different systems. I still kind of get to the mind of you got to respect the physiology, and the design of the system really needs to marry with the biology. If it's just engineered, you can miss some of the fundamentals and not be able to hit productivity for that reason. So those are key, being able to work with your engineering team when you're designing those components. I'll just kind of keep moving through here. So I can kind of I added a bunch of extra slides that kind of go beyond the advancement, but I did just want to kind of briefly go over that. Water treatment is key for us, and really advanced systems. We're looking to use ultrafiltration systems like you'll see here in our new facility, and that'll allow us to use dewater functionality, which is really important beyond live feeds. If you're doing high value co-products, or you're harvesting for other purposes between live feeds, beyond live feeds, that's going to allow you to basically dewater and then go to an extraction stage. These are really high energy and these are really costly endeavors, and that's where, commercially, that's where they're spending a lot of their costs beyond the production side. But that for us is going to allow us to do a lot more research in other areas beyond just aquaculture, live feeds. It allows us, bioplastics is what we're looking to work for, even within USM and some other projects. But there's a lot of opportunities once you get to having that capability. Commercial use, I'll just kind of point on this. Biomass production is kind of human consumption, biofuels, bioplastics. Biomedical is a big side of what algae is being used to these days, from pharmaceuticals to antimicrobials, cosmetics, colorants, water pollution, and then CO2 sequestration. I do say that with a pause, since geoengineering, this is one that is always, and it is becoming more into fashion. The iron enrichment experiments are kind of back on the fashion, but there is a lot of, I will couch that, and there's a lot of, I would say you need to look before you leap when you're doing things on a global scale. So that's one I will say with that. It's got a ways to go before I would say that really should be deployed. Algae biotechnology, beyond what we do here, which is live feeds, you can see our main purposes in-house are really oyster production is probably our major end user right now for our oyster larvae production and broodstock conditioning. And that is both for restoration as well as development. We're going to be involved with NOAA Center now for really developing new oysters that are more resilient in the future, so that we can work towards recovering some of these natural oyster beds offshore. So that's probably the biggest one we've been working a lot with over the last, I would say, 5 to 10, probably 10 years now. The high value co-products I do like to mention beyond, like I said, the live feeds, nutraceuticals, biofuels, pharmaceuticals, chemicals, cosmetics. Those are all big industries that algae does meet the demands for in a lot of different ways. I kind of added this one in here. This was a good from the literature. I think it does a good representation of what purposes you can use them for. Up on the right, it's probably closer to where you're seeing in aquaculture. Wastewater management below on the left, and those really have to feed into biofuels or some other avenues. And then really the nutraceuticals or the pharmaceutical industry really feeds, and that's where it really gets cost prohibitive, in that you have to have really high quality to do that. So, okay. The last here, just to kind of close things off, I did want to kind of just leave you with some of the areas we're interested or we're currently working in. That is strain development with algae as far as live feeds production goes for the Aquaculture Center. We do like to currently investigating microbial dynamics and really, a lot of different avenues there as far as the microbiome within systems, both within the algae culture and the downstream itself. Nutrient composition, optimization, that goes in pairs with that, and then crop protection, as well as investigating algal microbial interactions. The last side is what's new to us is really developing macro algae in IMTA projects. So that's what we're currently working towards now in innovating there. And then I kind of touched on this throughout. So we have a new facility that we're currently going to be building out over the next year. That is going to give us a lot more research capabilities. We will be bringing in more staffing for that and a lot more opportunities to work. We also have an offshore project that is starting off this year, and will be involved in supplying both the finfish program, the oyster program, and the algae program, supplying seaweed, oysters, and I think it's red drum that we're providing for that. And then lastly, just to kind of let you guys can push any interest you have to our director. He can field the broader interests. But we do, have opportunities. I know we have a summer internship program. That's usually Alcorn State in Tuskegee that we've been involved with. We do a lot of part-timers with undergrads and I guess the community college here as well. I've included the USM employment website, since that's where we post a lot of our new positions, and we do have positions continually open. I will definitely push those towards Amanda so she can relay that on to everybody as those come up in all our areas. But I think that kind of closes it out. And if anybody has any specific interest with algal cultivation, they can send stuff directly to me, and I'd be happy to field that.
Thank you so much, Egan. I do have a very quick questions because I want to be mindful for our next speaker. We are getting into scaling up, and of course, that's something that's financially constraining for a lot of community colleges to work with big old bioreactors. We are starting, I think, with, and correct me if I'm wrong, Claire, if you're on here still, we're going to do 5 liters and 10 liter scale ups. Is that a good experience for the students to eventually go into a larger facility? Because it's just so hard if you're not at a area that provides those types of opportunities, which is usually industry.
Yeah, like I said, a lot of R&D can be done at the 1 liter level. There's a variety of, systems that are set up there. I think, we use different vessels. The glass carboy, the 12 liter, the 12.5 liter, we use that as that's our workhorse, both because it's big enough that you can kind of represent what some of our PBRs, the light path, because as you get bigger, you can't get penetration into the culture with self-shading, but it does allow you to kind of, operate that semi-continuously, and even continuously, you can just use it as a chemostat. So I think that stage is a good stage, and you can learn, the techniques at that stage very, very effectively. Yes.
Well, thank you so much. If anyone has any questions, you can put them in the chat and I'll make sure that Egan gets them. And this leads us to Dr. Ian Rambo. We apologize for running late, everyone, but we appreciate your patience. Dr. Rambo is a marine microbiologist whose research focuses on interactions between viruses and microbial communities in marine environments, and his work bridges marine microbiology and applied biosciences. And this contributes to both fundamental scientific understanding and practical applications for food safety and allergen diagnostics. Welcome, Dr. Rambo.
All right. Hey, thank you so much, Amanda. Appreciate that introduction. And thank you all very much for having me today. It's a pleasure to be here. So let me just make sure we can see the slides. Sorry, just get the sheet. All right, y'all. Are my slides coming through?
Yes, we can see them.
All right, perfect. Okay, good stuff. Well, yeah, again, thanks so much for having me. I'm Ian Rambo. I'm a postdoc with the USDA Agricultural Research Service. And like Amanda said, my background is in marine microbial ecology. We're touching a little bit on that today, but what I'm going to talk about is my current research on applied artificial intelligence for peanut and tree nut allergy diagnostics. So first off, very nice to meet you. In terms of my journey through my academic career, I did my undergrad at the University of Rhode Island. First got into marine microbiology with David Smith out of the URI GSO, worked on persistent organic pollutants and the ability of microbes to degrade those POPs. Did my master's with Jennifer Biddle in Lewis, Delaware at the University of Delaware. Then I went to Texas. I lived on an island in the Gulf Coast for about 5 years with Brett Baker, and his lab was focused on marine microbial ecology, but a lot of evolutionary biology. I also spent some time out in Berkeley at Lawrence Berkeley National Lab working with Susanna Tringe on a virus microbe project centered around mangroves. I'm currently working with Sohaila Maleki at the Southern Regional Research Center in New Orleans. Her lab specializes in peanut and tree nut allergies. I also work with Adam Rivers out of Gainesville, Florida. So he's the artificial intelligence side of things and also works on microbial genetics. So we both worked with Susanna in the past. And as I mentioned, I'm an ORISE postdoc in bioinformatics and statistics. And I've been there since June 2022. Unfortunately, my time is likely going to be cut short at the end of May this year due to Doge activity. So looking for the next adventure. So I'm excited for that too. Opportunities, right? But I currently live in Alexandria, Virginia with my wife, Laura. She's an active duty Coast Guard, and my daughter, Veronica. So outside of science, I'm also a musician. I've led an Irish folk band. I'm currently doing I call it psychedelic doom folk metal project. Pretty excited about it. But I love going in the outdoors, cook a great shrimp etouffee and a gumbo. I'm from New Orleans originally. That's where I grew up. So I always love coming back. My family's still there half the year. So yeah, when I can make it back to Jazz Fest, it's a good time. So yeah, if you've ever seen Kindergarten Cop, I might be dating myself, but yeah, it's like, who is your daddy and what does he do? Well, let me tell you what I do. I'm a computational biologist. So what does that mean? I know we hear these days, okay, data science, that's really an exciting thing. I'm combining a lot of different tools, a lot of ways to visualize data, to analyze data. Some of you might have used these in your classes, things like, scikit-learn or NumPy. But my other side is bioinformatics. So I like to use data to solve biological questions. And looking at this schematic here, I pulled this up today and I was like, oh, I've actually done most of these things. For instance, with the microbial ecology side of things, taking DNA sequencing data, being able to reconstruct genomes of those uncultured microbes, and then figure out where they fall on the evolutionary tree of life by looking at their proteins. So it's those types of questions that, as students can start to think about, okay, how can I solve this certain problem or question using these certain tools? And what we're going to be talking more about today is the disease prediction side of it. But in terms of some of my projects I've worked on, and if you want to connect with me on LinkedIn and talk about them some more, I'm not going to dive into them today because of time, but I'd be happy to talk about them with you some more. But I've looked at things like, if you're familiar with epigenetics, right? There's something called DNA methylation that might be used to regulate the genomes of microbes in the environment. I've also asked, okay, what are different antimicrobials that might be used as antibiotics in nature? For the algae people out there, I've looked at bacteria that live on toxic microalgae called Gambier discus. And using those methods where you can reconstruct a genome, you can ask who are they and what can they do? What are their functions that they're capable of? What I've worked on most recently in the microbe side of things are viruses of a group of archaea called asgards. And these are our closest prokaryotic relatives. So we got samples from, like, was it 2,000 meters below the sea? Bottom of the ocean, and very cool communities there. And what I'm finishing up with my lab when I have the time, is looking at virus-bacteria interactions and how they affect biogeochemical cycling in mangroves. But let's get into my main projects that I've been working on at USDA. So I was looking at how do patterns and antibody binding to peanut proteins shift after allergic people take a certain treatment to treat their peanut allergy. And what I've been working on with my our postdoc Lauren and the rest of the group is how can we use certain cross-reactive protein regions. Say if somebody has a peanut allergy or walnut allergy, why are they allergic to the other proteins in that other, say I'm peanut allergic, but I'm also allergic to walnuts. Why is that? You can have regions that are cross-reactive, and these might be used as diagnostic areas that we can look at. I've also done a little work looking at salmonella pathogenicity, bit of work with working on large language models for batch effects correction with 16S microbiomes. But what I'm going to be talking to you about today is looking at how can we improve food allergy diagnostics using computer vision. So using machine learning, but deep learning. So let's get ourselves situated in terms of the problem that's being faced here. So there's a food allergy epidemic going on in this country. We've got 33 million Americans with food allergies. Some of you might have food allergies out there. You might know somebody with food allergies. I have several, family members and friends that have food allergies. It's difficult. And it also can be a very expensive thing in terms of, emergency room visits, specialized diets. So it takes a lot of economic toll. And only 10 to 20% of people will outgrow those allergies. On top of that, we have underrepresented populations are at a higher risk, often due to living conditions, and they often lack treatment options. Not a lot of allergists in the areas where those populations are. So we also need to think about how we can make healthcare more equitable to all Americans and hopefully, people outside of our country too. So these days we're always hearing AI this, AI that. There's the artificial intelligence revolution is changing the face of healthcare. It's changing the face of how we do science. So how can we harness this tool in the field of food allergy? So let's just get ourselves a very tree level view here. When I'm talking about an allergic reaction, this is something called an IgE mediated reaction. So the IgE antibody, you can think of it as the antibody that's driving the allergic reaction that's caused. There's also another antibody, IgG, in this case IgG4, that we can think of it as kind of suppressing the reaction. It's a very simplistic way to look at it, but that's really what we need to think about going forward. IgE causes the reaction, IgG4 will help to negate the reaction. So in terms of treatments, I had mentioned I looked at ways that if people who have peanut allergies can use certain therapies to gain tolerance. So one of these is peanut oral immunotherapy, where essentially you're eating peanuts over time and gradually increasing your dose. Peanut sublingual immunotherapy is you're using purified proteins under your tongue. So these are things that are being looked at. One of them is FDA approved, called Palforzia, that Sohela's lab has worked with. And these are promising treatments. But in terms of how we diagnose allergy, you might have had these done to you. I had it done too, with this, prick your skin. I had the itchiest back for like days. It was not fun. So it's also, time consuming and not always very accurate. Measuring the IgE in your blood, your sera. But the gold standard is known as the oral food challenge. So that's essentially you're given the food and you wait and see if you have an allergic reaction. So it's the most reliable way, but it's also very expensive, time consuming, possibly dangerous. And you have to consider if you are a single parent that has a lot of time constraints, you are spending multiple hours out of your week just doing these oral food challenges. So we can do better. So in terms of the sphere of AI things, let's kind of consider this onion figure here. So artificial intelligence is a very big blanket term. But we have things like machine learning, and there's classical machine learning, where we have models that are able to learn from more data over time. So in this case, I've used this to predict peptide biomarkers, so parts of the protein that seem to show peanut desensitization. But what I'm currently working on is the deep learning side. So this is where we get into multi-layered neural networks that can learn patterns from large amounts of data. So I'm using a technique called supervised machine learning. There is unsupervised. I'm not going to go into that today. But in this case, we're teaching an algorithm and by showing it examples and telling what each example is. So let's look on the left side. So we have a training set where we have, say, 1 or 0 being allergic or not allergic. The algorithm can see, oh, allergic looks like this. And once you've trained it, then give it an unseen test set or validation set. And from that unseen data, we can see how well the model performs. But you have to be careful with how your data are set up, because look at this comic here. It's a bit silly, but okay, you show a child daddy, you have the plumber's crack here. The child might think daddy is, say, 2 melons at the store or 2 balloons. So you have to be careful in terms of how your data are labeled. Garbage in, garbage out. That's something really to consider if you're doing anything regarding data in science, in terms of your data stream. But in terms of framing those food allergy classification problems, what I work on and kind of how I've guided my time here at USDA is thinking about, okay, what kind of diagnostic issues and problems can we address through this? One of these is binary classification, where we might want to say, is someone not allergic or allergic to peanut? Another is multi-class. So between peanut, cashew, walnut, or pistachio, we might want to know which one of those 4 are they allergic to. So let's say this person's allergic to peanut. Then we're getting into the tricky areas here is multi-label, where we might want to say the person is peanut and walnut allergic. So right now I'm focusing on binary classification. Start simple. So in terms of this, the goal of my research is to develop a safe, accurate, and non-invasive test that can match or exceed that oral food challenge gold standard. I need to match or exceed the accuracy and precision of it at substantially less cost and time. So what I'm utilizing here is a computer vision approach. So I'm taking linear peptide, IgE, and IgG4 binding data. So these are pieces of those allergen proteins, and having the IgE, remember the driving antibody, and the IgG4, the, say, reaction reducing antibody, seeing how those also interact with the physiochemical properties of the protein, and then overlaying that onto 3D protein structures. And often we have issues with small data set sizes, especially when it's clinical data of rare patients like food allergy patients. So I've been exploring data augmentation techniques. These are ways that you can use smaller data sets to do something called fine-tuning with a model. That's to say, like transformers, like ChatGPT, for instance. These take massive amounts of data to train. Once you've trained it, then you can fine-tune it with a smaller data set size. So that's the approach I'm taking here. So in total, Sohaila's lab has gathered over 1,000 different peanut and tree nut allergic participants from 7 countries, I believe. We have clinical metadata and oral food challenge labels. So we have those strong oral food challenge labels, which is really key. And what I'm doing through this project is binary classification of peanut allergy. The thought is that getting at least a better peanut allergy diagnostic algorithm out there and for the general use, that's something that I'm hoping can better the quality of life for the people. So brings up the question, okay, is this a simple problem and I'm applying just a very complex tool to this? Well, yeah, allergy diagnostics are not as simple as I as I anticipated coming in. So there's a lot of different factors taking place where deep learning can be used effectively. But as I mentioned, I'm using something called a vision transformer. What this does is, say you have an image. In this case, we want to classify whether you have a dog or a person in this image here on the left. What it does is it splits a image into different patches. And essentially, we're looking at the relationship of those patches to each other, and those are fed into a classifier. And in this case, we can say, okay, there's mainly a person and a dog that are seen in this picture. But kind of showing, this is mock data, mind you, but for one of the major allergens, ARH2, this is a major peanut allergen. What I'm doing here is a method called cube map representation, where we have a 3D allergen, and think if you have it suspended within a cube. I'm looking at those 6 faces of the cube, and this gives the model a much more robust data set to look at. And then from there, I'm splitting those images up into those different patches that will be fed to a vision transformer. The thought is here, I can use something called the self-attention mechanism of a vision transformer. So as I mentioned that, and if you've also, think about this, if you've used any thing like ChatGPT or Copilot, when you're creating those text prompts, have you ever tried giving it more context of being like, you are an expert in algae, you're an expert in guitar pedals or something. Whatever you want to do, you can give the model more context and then it can adapt accordingly. The same thing goes for the images. So the way that's working is something called self-attention, where it's able to learn how these different parts of the data interact with each other and weigh the importance of those patches against each other. So then you can visualize it. And the hope here is that this will allow clinicians to better understand how the model is performing. Because I have heard clinicians say, I would never use AI in the clinic because it's a black box. The fact of the matter is that being able to explain the models is a really big part of research in the machine learning field. So if you're using models like this in your research at all, definitely make sure that you have explainability methods. And that's something I really try to strive for. All right, so some takeaways. IgE-mediated food allergies, they're on the rise. And our current diagnostic methods are not entirely accurate. They depend a lot on the individual, but the gold standard is time-consuming and expensive. So what I'm doing at USDA is developing this vision transformer approach with this large data set that's been collected over a decade, and using this to develop a diagnostic algorithm for food allergy. So, hey, thanks so much to the Molecular Lab, and this is actually the better acknowledgements page. The Molecular Lab out of New Orleans, fantastic people, so great to work with. I'm very thankful for everything that I've gotten to do with them. And yeah, I'm going to do the best I can to get this project together before time runs out. And thank you very much, Amanda, for having me, and Jackie for nominating me. For those of you who worked with Dr. Nesbitt or taken her classes, yeah, she's wonderful, and I've known her for a while. So it's yeah, I was very, very happy to come and share my my research with y'all. So for that, I think we have some time for questions. So I'd be happy to hear what you have to say. Thank you.
Just for, yeah, the memes have been great. Just for the students on here, on Dr. Rambo's previous, I think 2 slides before, one of Delgado's students has worked on the project with them
Yes.
also, Angela Graham. So she is graduated and now pursuing a bachelor's while still working at the USDA. So thank you. Does anyone have any questions? Dr. Rambo, just because of where I feel the world is going with data analysis, bioinformatics, the big data push, students are hesitant to take more math classes than required.
Mm-hmm. I understand. Yeah.
What would be your, I guess, encouragement, feedback, advice to help them overcome this just generalized uncomfortableness around math?
Yeah. Well, I'll just tell you this from a personal experience of myself, is that as a younger man, even now, I always have this thing of like, oh, math, I'm not good at math. And I think a lot of that just held me back. So I think part of it is that often we're trying to teach math in a way that's supposed to fit everybody, but it only fits certain people. Think about how your brain works, how you learn best. Because I found when I approached calculus in a different way and saw like, okay, I found a great book and I can share it. It's an older book, but the wisdom still holds, is that, you don't need to overcomplicate this. When you actually break it down into simpler language, it's not as complicated as you think, because then you start looking at like, oh, this equation just means this. Because at first, yeah, it's a bit, oh my gosh, what the heck does this mean? What's this symbol? So what I would say, to get started, you don't need to be an expert. You don't need to be a math whiz to get started in machine learning. You can at least, learning Python, learning how to use these libraries is great. where it does help is at least trying to understand how the models work. So here's where I'll say too, what I found really helpful is if you're using, say, any of these like ChatGPT or Claude or things like that, large language models, they're great educational tools. Because I've tried this type of prompt where it can say, I need to understand how the self-attention mechanism works for a vision transformer. Explain it to a layperson, explain it to a 10-year-old. Now explain it to a PhD in data science. So you can get those different layers. And that's helped me tremendously of like being able to just peel back the layers and then understand how the math works. So yeah, hopefully that got around to the question, but yeah, I know taking math courses can be difficult. But if you can take the time to learn it and get the foundations of these models, it really helps down the line. Yeah. But you don't need to do it to start.
Well, thank you so much. I also, we also have two other students that have worked on this project. Also, Zanab, who is still working at the USDA, specifically in the lab with the SRRC, and then Gregory. So,
Oh yeah.
Yeah, you've had a lot of impact on our students as well as.
Sorry, Gregory. I was like trying to get my acknowledgements slide together last minute because I was like, God, geez. So yeah, shout out to Gregory too, because he's done excellent work with Sohaila's lab, and yeah, everyone is up.
Well, I appreciate your time and your dedication and your commitment to helping us and giving our students some more perspective. Just as a reminder, if you haven't filled out the survey, please do so. I placed it in the chat just really recently. And then I will make sure to get out the presenter PowerPoints that do share with me and information about them as well. And then last, Dr. Rambo, we do have one last question. How will AI affect the field of bioinformatics in terms of making it more or less obsolete? Is it worth taking the time to learn programming languages like Python?
Oh yeah, that's something I definitely considered. Yeah, because yeah. So here's the thing: is at least my thought on this. AI is increasing at such a rate that soon it's going to have the capacity of, 1,000 PhDs. Think about that. I think what you need to consider is the human side, I don't believe is going to become obsolete. Think of it as a way that we can, if we were able to work with it effectively, we can increase the amount of research we're doing and the effectiveness of the research we're doing. So in terms of learning Python, though, I would say don't let that deter you. Because the thing is, in terms of AI writing code, it's very good at it, but it's not always going to be perfect. Because I've had times where, I wanted to try to optimize something that I've written, and it just spits back what I gave it already. It doesn't actually do anything to it. So currently at its state, it's not perfect. So yeah, I would say, yeah, I don't think it's going to make things obsolete, but stay on top of the trends because it's going to be a tool, ideally, that can be used to move bioinformatics at light speed.
We have one more question, if that's okay.
Yeah.
What AI course should students of biotechnology, chemical technology take, which would give them better insight or skills?
Yeah. So I don't know in terms of what's available to you at Delgado. What I would say first off is that one popular one is Andrew Ng's course out of Stanford. It's also kind of the thing of like, I took Andrew Ng's course on machine learning, and now I know machine learning. I've found that the updated version of the course is great because first off, it's written in Python. It's a language that if you're doing any sort of machine learning and algorithm development, that's what you're going to be using most of the time is Python. And what I found was really useful and this kind of brings us back to the first question as well, is that you didn't need to be a math expert. If you wanted to dive into it, you could, but you at least got an idea of how things worked. So check out Andrew Ng's course. I think Towards AI, I think is the group, but just look for Andrew Ng Stanford on Coursera. That could be a good place to start. In terms of like, I haven't actually taken any like official courses through university on AI. So a lot of what I've had to do has been just learning in the field, pretty much, of, okay, we have this problem. How do I get around and do it? How do I do it? So that experience has been useful. So yeah, so I would say check those out. And then honestly, start experimenting. You know, if you have some questions, see if you can apply these things, because that really, in my opinion, getting that real boots on the ground type experience is invaluable.
Thank you so much. People are really interested in asking a lot of questions, so this has been a great topic to kind of end with. If anyone, I keep saying this, but if you have any questions or things that you think about that you want to ask the presenters, feel free to email me or put it in the chat before we get off. This entire recording will be posted. The links will be shared out. They will also have chapters placed in there. So it'll be easy to navigate to each speaker. And then again, I will send out the presentations that are sent to me and share it with the team. Let's give a big round of applause for all of our presenters for all the time that they took to present and share their expertise and experiences and day in their life in the biotech, chemtech world. Well, thank you for everybody and everything that's been done today. We appreciate your time, and I hope y'all have a wonderful day, and thank you for being patient as we went a little bit over. And I'm throwing my information in the chat in case anyone needs to get in touch with me. And for the speakers that are left, we had a great attendance today. We had over 35 students come off and on throughout the day. And we had about a total of 50 attendees throughout the day. Thank y'all so much. And I will be reaching out to all the speakers probably this afternoon or tomorrow with just a thank you and follow up.
All right. Hey, thanks so much, Amanda. Thanks for putting this together.
Bye, everyone, and thank you again.
Thank you. Bye.